Good morning. Y'all look great. Is this not one of the very best days of the year for all of us? For how many of you is this your very first conference? Oh, a few of you. Okay. Well, really, welcome. You're going to have your socks blown to smithereens. This is really one of the best conferences in the country. And this is 35 years that this particular conference has been helping to bring together the greatest brilliant minds in Alzheimer's and dementia care and research. I have had the privilege of attending 25 years. For those of you I don't know, my name is Patty Mouton. It is my rare privilege to serve as vice president at Alzheimer's Orange County. I've been there coming up to 19 years. And before I came on staff, I volunteered for them for about 5 or 6 years. And so my affinity toward our cause, toward the people we serve, toward the brilliant clinicians who help the families who deal with these really terrible diagnoses, my hat is off to all of you. As the French say, chapeau. Welcome. I hope you have a wonderful day. It goes without saying that all of us here today, every single one of us, even those who did not have the great, rare good fortune to know him, we all owe a very deep debt of gratitude to Dr. Carl Kottman, who started this conference 35 years ago and actually performed some of the most foundational and earth-shaking research into our understanding of Alzheimer's and related dementias. So as we go through the day, remember Dr. Kottman and say a thank you, because I think from wherever he is, he must be very pleased and proud of every single one of us for carrying on this most important intellectual tradition. Thank you. Have a great day. And now, I'd like to introduce my colleague, Deborah Leigh from the association.
Thanks, Patty. Hi and welcome. I know you're going to be seeing a video soon as well about Dr. Carl. So I'm Deborah Leigh. I'm the executive director for the Alzheimer's Association Orange County chapter. It's really lovely to see all of you here today. You know, my grandmother once said that our health is our greatest wealth, and I believe that every single one of us in this room, around the world, deserve to live a life free of Alzheimer's and all other dementia. So the Alzheimer's Association is the leading organization when it comes to research. We are the only organization working to ensure that everyone in every community has the information, support, and resources they need to navigate every step of this journey. We're very proud to partner with Alzheimer's Orange County and UCI Mind to put on this conference today. You're in for a treat. I did want to just tell you a little bit about the Alzheimer's Association for those of you who aren't familiar with what we do. I'll just share 3 quick things. 1, we are supporting people living with the disease and their families. Number 2, we fund more Alzheimer's and dementia research than any other nonprofit globally. And number 3, we are the only Alzheimer's and dementia organization with a presence both at the federal level and in all 50 states. So just locally, to bring this closer to home, we're providing really vital care for individuals living with the disease and their families. We offer free support groups, education programs. The one thing I do want you to take away is our website, which is alz.org. And our 1-800 number, I think, is also really important for you to have as a resource in the community. If you've just gotten a diagnosis in your family and you don't know where to start, it's a great place to call. If you're in crisis and you need to call somewhere, it's a great place to call. If you need to find resources, it's a great place to call. The number is 1-800-272-3900. We're here today to listen to some really exciting research breakthroughs. It is a very exciting time to be in this field right now. We are the largest nonprofit funder of Alzheimer's research, and many of the important researchers who you'll hear from today have probably received some funding from the Alzheimer's Association through part of their career. In fact, we're, in the next few weeks, Josh, going to be giving a $500,000 Alzheimer's Association Research Grant Award to a UCI researcher. Yeah, we are currently investing over $3 million in this local community, over $63 million in the state of California, and globally $450 million in Alzheimer's research. Our investment today will lead through breakthroughs tomorrow. And finally, I just want to briefly touch upon our advocacy work, because we are working both at the federal and state levels. In the springtime, we have about 1,000 volunteers who come with us to Washington, D.C. They share their stories to urge lawmakers to make policy changes and fund research when it comes to dementia care and for new policies. And then in the spring, we also go to Sacramento. So if you're wanting to share your story, please see one of our team members who are wearing purple. They will help you get involved with us, whether it's you need resources, if you want to join us as a volunteer, providing education, or coming to us to DC or Sacramento. So you're in for a treat today. Thank you for being here and investing your time and learning, and to the researchers who are going to be speaking today. It's now my pleasure to introduce the director of UCI Mind, Dr. Josh Grill.
All right, good morning. Thank you all for being here. We're very, very excited about today's conference. As you heard, it is our 35th. I am Josh Grill. I'm going to be your host today, which means I am obligated to get a couple things out of the way before we really get going, like the fact that the bathrooms are outside and down to your right. I really want to encourage you to visit all of the booths for our exhibitors here in the room and out in the hall. This conference is made possible by their participation, so we're very grateful to them for being here. If you haven't, we really need you to check in at registration. This will give you your badge and perhaps more importantly, your parking validation, and you can pick up the program. If you're a person who's pursuing CEUs or other units today, that we especially need you to sign in to get those credits, and we need you to do that by 9:15 in the morning. We do have a fantastic program today. We have brought together some of the very best minds from around the country and here at UC Irvine to deliver presentations on incredibly exciting and important topics. We're so proud to be able to do that. We're so proud of our mission at UCI Mind and proud of our partnerships with the Alzheimer's Association and Alzheimer's Orange County. I'd be remiss if I didn't thank our partners who really help us put this event on. You heard from Patty Mouton, but also Lauren Weiss and Jim McAleer from Alzheimer's Orange County who helped us plan this event. You heard from Deb Levy, but also Jennifer Maurer from Alzheimer's Association. From the UCI Mind side, I really want to thank Megan Whiprock, Dr. Megan Whiprock, Sasha Stone, and Shirley Siravong, among others who put a lot of time and energy into planning this event. That time and energy is because though the crux of what we do at UCI Mind is about researching ways to find solutions for people dealing with these diseases, we really fully embrace the aspect of our mission of bringing the information back to the community at large so that others can benefit as quickly as possible from what we're learning. And again, today's theme is particularly exciting. It's about how research is changing clinical practice and the progress that is being made. And so we're just over the moon to be able to talk about new approved drugs and new methods for helping families navigating the odyssey of having a loved one dealing with a cognitive disorder. The fact that These things are being paid for by agencies in this country. It's an incredibly exciting time. I am confident that you will also walk away from this conference understanding that there is a lot of work left to be done. So it is a unique juncture in our history as a field, an exciting one, but we are not lightening up on our efforts to help the millions of people who are dealing with these diseases. As you heard, it is our 35th annual conference, which I think is an incredible achievement unto itself. We survived the pandemic, kept this conference going annually, and as you also heard, it would be tragic to not acknowledge the person who started this conference 35 years ago, Dr. Carl Kottman. Dr. Kottman was the founding director of UCI Mind. He was the founding director of the UCI Alzheimer's Disease Research Center, which was one of the first 5 centers funded by the National Institute on Aging in 1984. As you heard, he is the source of tremendous discoveries in our understanding of the aging brain and the brain afflicted with diseases that cause dementia. He started this conference. From the beginning, he recognized the importance of bringing what we're learning from our work back to the people we are trying to help. He was foundational, he was inspirational, he was of major impact to our field, to all of us at UCI Mind, and I expect to many of you. I would like to take just a couple of minutes to honor Dr. Kottman's legacy with a short tribute video.
An exceptionally gifted scientist, a tremendous builder, a generous mentor, an intuitive risk-taker, and a scholar who was dedicated to transforming human health. These acclaims and more describe Carl Kottman, Distinguished Professor Emeritus in the Departments of Neurology in UCI School of Medicine and Neurobiology and Behavior and the Charlie Dunlop School of Biological Sciences. Carl was an early and constant leading light in the fight against Alzheimer's and other neurodegenerative conditions that cause dementia. Key to building UCI's world-class stature, he also inspired researchers around the globe. Professor Kottman majored in chemistry at the College of Worcester and earned a master's in analytical chemistry from Wesleyan University. He received a PhD in biochemistry from Indiana University in 1968 and joined UCI the same year as an assistant professor in the Department of Neurobiology and Behavior. In 1974, he became a full professor in the department, and then in 1983, a professor in the Department of Neurology. Carl was the original director of the UCI Alzheimer's Disease Research Center, which began in 1984 in partnership with USC and was one of the initial 5 centers in this prestigious network of more than 30 centers today, thus launching a prominent and distinguished history in dementia research at UCI. In 1995, Carl became the founding director of UCI Mind, which he went on to lead for over 2 decades. UCI Mind was established to pursue interdisciplinary investigations of both pathological and normal neurobiological changes that occur as the brain ages. As a multidisciplinary center combining basic science and clinical research, UCI Mind conducts research within the context of its ultimate goals: to develop methods for preserving normal brain function, conserve or possibly restore normal brain activity once damage has begun, and engage in the pursuit of effective therapeutic treatments. Carl's research significantly advanced our understanding of Alzheimer's disease and related neurodegenerative conditions. His unflagging curiosity sparked his research. When he started wondering why the aging process seemed slower in physically active people, he set out to investigate. He studied the effects of exercise, diet, and other lifestyle factors on brain health, which has implications for Alzheimer's disease prevention and management. His contributions to the field include exploring how these factors can influence neuroplasticity and cognitive function, aiming to develop strategies to mitigate or prevent Alzheimer's disease and other forms of dementia. His exploration led to his celebrated discovery that exercise significantly increases the protein called brain-derived neurotrophic factor. This finding led to clinical trials of exercise for people with mild cognitive impairment, and numerous others continue as guideposts for neuroscientific research into aging and age-related brain disease. His illustrious career resulted in the publication of 11 books and over 800 articles on multiple facets of neuroscience, including 21 papers in the eminent journals Science and Nature. A long list of honors was bestowed upon him. Among them are the Alzheimer's Association International Conference's Lifetime Achievement Award in Alzheimer's Disease Research and the National Institute on Aging Leadership and Excellence in Alzheimer's Disease Award. Other recognitions are numerous and included the 2004 UCI Medal for Dedication in Research, Teaching, and Community Service. In 2003, 3.5 decades into his storied career, the journal Neurochemical Research dedicated an entire edition to him and his achievements. It included articles by his peers, including former students. One author observed that Carl's ability to make the direct transition from graduate student to independent investigator was a clear early indicator of his extraordinary talent. Another said, Carl has been an influential leader in advocating the mining of neuroscience for medical benefit. Still others singled out his enthusiasm for science and his ability to transfer this enthusiasm to his coworkers, his willingness to take risks, openness to fresh ideas, and that he was ahead of the curve, a distinguished basic neuroscientist pushing the translational envelope before doing so became popular. We will forever hold admiration for this colleague and friend who, even amid his many accomplishments, was always excited about the newest possibilities. Carl told attendees at a SoCal Alzheimer's Disease Research Conference that science doesn't stay quiet for very long. Professor Kopman's influence will continue to speak to us as we move forward on changing the future for the better.
We lost Carl in August, but we all stand here because of him, and we all will carry forward our research mission in his honor and built upon the foundation that he built here at UCI Mind. So I'm going to move us forward with the day's agenda, which is incredibly exciting and really does highlight the progress that we are making in trying to change clinical practice and change people's lives for the better. You're going to hear from fantastic, illustrious speakers from around the country, and we are thrilled to welcome Dr. Aimee Pierce as our first speaker. Dr. Pierce is a professor of neurology in the School of Medicine at the Oregon Health and Sciences University in Portland. She did her MD at Columbia, her residency at UCLA, and her fellowship in dementia at UCSD. She is the Director of Clinical Care and Therapeutics Program at OHSU's Latent Aging and Alzheimer's Disease Center. She's been the PI of numerous trials, including many when she was on the faculty here at UC Irvine. So I'm sure she is familiar to many of you. It's a great pleasure for us to offer a homecoming of sorts for Dr. Pierce, and we're thrilled she's accepted the invitation to talk to us about, among the most exciting progress that we have seen in recent years, newly approved drugs for the treatment of early Alzheimer's disease. Dr. Pierce.
Good morning, everyone. And I really want to congratulate UCI Mind and Alzheimer's Orange County and Alzheimer's Association for 35 years of this wonderful conference. And really thank Carl Kopman for his vision and leadership in the field. And it is truly an honor for me to be back here in Irvine, where I started my career in the field of clinical care and research for patients with Alzheimer's disease. And I do have a few disclosures. I am a site principal investigator for numerous Alzheimer's disease clinical trials sponsored by pharmaceutical companies as well as the National Institute on Aging. And I take care of patients as well as conduct research on Alzheimer's disease treatments. And so it's a very interesting time. I'm sort of bridging that gap between clinical trials and patient care right now. So for many decades, the hallmarks of Alzheimer's disease were only able to be detected after a patient died. But even with the naked eye, we could see that patients with Alzheimer's disease had severe shrinkage or atrophy of their brain compared to people who died without Alzheimer's disease. And under the microscope, we could identify amyloid plaques as well as neurofibrillary tangles comprised of tau, these dark-shaped structures. However, being only able to diagnose Alzheimer's definitively after death really limited the ability to make extremely accurate diagnosis during life, and also affected the ability to conduct clinical trials and finding the exact right population to test. So in the past bit more than a decade, there have been major breakthroughs in Alzheimer's disease diagnostics, and now Alzheimer's markers can be seen in living patients. This has a major impact on diagnosis as well as developing treatments that are helpful. And that involves PET imaging, which can detect amyloid and tau, checking cerebral spinal fluid by a lumbar puncture, looking for the markers of Alzheimer's, and most excitingly, blood-based biomarkers that can detect amyloid and tau. And we'll be hearing more about that later today. This is an example of an amyloid PET scan. On the left, showing a brain of someone with Alzheimer's, and on the right, someone without. And indeed, the amyloid plaque is quite visible in red and yellow, deposited throughout the brain. So when thinking about treatments for Alzheimer's disease that may hit the target, it's important to consider the amyloid cascade hypothesis. And this is rather complex, but the crux is that the deposition of amyloid in the brain is early and critical in the development of Alzheimer's disease. And this has been a hypothesis in the field for many years, and evidence has been accumulating, and now some of the clinical trial evidence is supporting this as well. And this shows that amyloid beta is generated by amyloid precursor protein by enzymes, and then amyloid beta production can lead to amyloid aggregation, inflammation, tau, tangle generation, excitotoxicity, and other oxidative damage. And all of these processes then affect the health of neurons, moving from healthy neurons to cell death, effects on neurotransmitters, and then ultimately what affects the patient's cognitive and functional decline. So in thinking about how to target amyloid and the amyloid cascade, one approach would be utilizing the body's immune system and immune mechanisms. So this is an important concept that we're all fairly familiar with. There are different forms of acquired immunity, including active immunity after an infection or vaccination, or passive immunity, either naturally, for example, mother to baby through breast milk, or passive immunity artificially through providing antibodies as a medication or treatment. And how could this be applied in Alzheimer's disease? So interestingly, natural antibodies to amyloid are found in the blood of people with Alzheimer's disease. And this may be part of the mechanism of our own bodies responding to amyloid buildup. Vaccines to amyloid and have been tested many years ago. They were a little bit too strong, causing encephalitis in the brain, but now new vaccines are under development and under testing for amyloid and tau, which may hit the target better. And then in terms of the passive immunity approaches, antibody therapy is what we'll focus on now. And the new treatments that I'll be speaking about are antibodies against amyloid, although tau antibodies are also under development. So anti-amyloid treatment, there can be antibody approaches, but also approaches that reduce amyloid production or other aspects of amyloid metabolism. But this graphic is quite helpful. It kind of shows that antibodies essentially fit with structures in the body with like a lock and key mechanism. So each antibody is very specific to its target. This shows that as amyloid is generated, it starts as a single peptide of around 40 amino acids. Then it can group together into protofibrils or longer fibrils, and then ultimately can deposit in the plaques that can be seen under the microscope. And each of the antibodies that have been under development bind to a specific form of amyloid. And what you can see is that each antibody is different, but it seems that the antibodies that have been showing most success tend to bind to the larger forms of amyloid that's aggregating. And antibody trials have been negative in the past, but do keep in mind that each antibody is different. And the current trials, which have shown some benefit, tended to select participants more stringently, treat patients earlier and longer, and then again, have their own special confirmation. So this has led to FDA approval for anti-amyloid antibody treatments, including aducanumab, lecanemab, and donanemab. I'm not going to speak much about aducanumab today, and that is because the data that supported aducanumab, there were some controversies based on the early closure of the study, the analysis of the results, the FDA approval, and actually the company that produces aducanumab has now removed it from the market, so it's not really available for patient care. But we will now talk about lecanemab and donanemab. So lecanemab is a humanized monoclonal antibody that binds to soluble amyloid beta protofibrils. And the study that led to FDA approval was a phase 3 trial, 18 months of treatment, randomized, double-blind, placebo-controlled, meaning half of the participants received active lecanemab and half received a saline solution intravenously. It was nearly 1,800 participants with early Alzheimer's disease with evidence of amyloid beta on their PET brain. And this is an example from an earlier study of lecanemab. This was an example of an amyloid PET participant before treatment and then after treatment with lecanemab. And you can see that there was a reduction of the red and yellow areas of the brain, which represents the amyloid plaque. And then graphically, we can see in this study, the placebo group is in blue and lecanemab treated group is in orange. And over time, the placebo group's level of brain amyloid held steady over 18 months, but the group that was treated with lecanemab significantly declined. So it was clear from this and other studies that lecanemab does reduce brain amyloid plaques. But in terms of what really matters to patients, and that's memory performance, cognitive performance, and functional abilities. And by functional abilities, we really mean how a person is taking care of themselves, taking care of their daily living tasks. Indeed, in this study, lecanemab did slow progression of this cognitive and functional decline in patients compared to those who took placebo by about 27% over 18 months. So the outcome that was looked at was called the CDR, which is an extensive interview with participant and their study partner looking at cognitive performance and daily living skills. And you can see over the 18 months that the lecanemab group in orange, it did decline, but it declined slower than the placebo group. And again, that difference was about 27%. Now, whether this 27% is very notable or meaningful to patients and their family members, that there is some discussion about that still at this point, but it certainly was statistically significant, and it was the difference was evident. However, there are some significant adverse effects of lecanemab as well as other anti-amyloid antibodies. Number one, infusion-related reactions. So this medicine is given by infusion intravenously. generally in an infusion center, or can be given by home infusions. But it's given by medical personnel, and one reason is the infusion-related reactions. And so about 26% of patients who received lecanemab had these, generally after the first 3 infusions and generally mild, but they would typically be like fever, flu-like symptoms, but could be more severe like blood pressure change or drop in oxygen level. Headache was also seen in about 11% of patients receiving lecanemab. But the most concerning risk or side effect with lecanemab and other anti-amyloid antibodies is ARIA, amyloid-related imaging abnormalities. So there are 2 forms of ARIA, ARIA-E, which means edema or swelling, and ARIA-H, which means hemorrhage. And here we can see the appearance of the ARIA-E and this white area in the brain MRI. And here we see little spots of black, which are demonstrating ARIA-H, a form of ARIA-H called microhemorrhages, which can be very small hemorrhages. There are other forms of ARIA-H, including large hemorrhages, as well as superficial siderosis, where blood can coat the surface of the brain. So overall, ARIA occurred in 21% of patients on lecanemab, compared to 9% in placebo. However, the majority of ARIA was not symptomatic, but symptomatic ARIA occurred in 3% of patients on lecanemab, and symptoms generally resolved. When symptoms occurred with ARIA, it would typically be headache, confusion, visual change, dizziness, nausea, gait difficulty, or focal neurologic deficits, like similar to a stroke or seizure. And the monitoring for ARIA is done by MRI, because in many cases it can be asymptomatic. And if it's detected, then treatment can be adjusted or stopped or paused. In terms of ARIA, it is really important to know that the risk is significantly higher in ApoE4 carriers. We know that ApoE4 is a genetic risk factor for Alzheimer's disease as well as for ARIA. And there are 3 possible variants of ApoE, 2, 3, and 4. We get 1 copy from each parent. And the highest risk of ARIA is in people who have 2 copies of ApoE4. And then a mid risk for people with 1 copy, and lowest risk for people who don't carry the ApoE4 gene. So for any ARIA, those with 2 copies of ApoE4 have a risk of 45% on lecanemab. 1 copy, a risk of 19%, and non-carriers, 13%. And the risk for ARIA with symptoms and serious ARIA is also higher as the number of ApoE4 genes increases. So with those caveats in mind, lecanemab was approved by the FDA in July 2023, a bit more than a year ago, for treatment of Alzheimer's in an mild cognitive impairment or mild dementia stage. Patient selection involves checking for amyloid positivity by a PET scan or cerebrospinal fluid, checking ApoE genotype so that counseling can be done about the risks of ARIA, a brain MRI within 1 year prior to check for other exclusionary findings like more than 4 microhemorrhages at the beginning, which would make treatment less safe for patients. Use of blood thinners warrants caution in these patients, and many clinics are not treating patients who are on blood thinners with antibodies. And the dosing is weight-based, 10 milligrams per kilogram of body weight of patients, infused over 1 hour every 2 weeks. So donanemab is the second antibody, which is now being used. There's some similarities and some differences with lecanemab. Donanemab binds to forms of amyloid beta only present in the plaques, so not the protofibrils. The donanemab trial that led to its approval was also 18 months, randomized, double-blind, placebo-controlled, nearly 1,800 participants as well, with early Alzheimer's disease who showed evidence of the amyloid on their PET scan, but also evidence of tau on their PET scan. So that was an added wrinkle there. Interestingly, amyloid PETs were done at 24 and 52 weeks during the study, and this allowed early switch to placebo if patients showed reduction of their brain amyloid before the end of the study. And indeed, donanemab cleared amyloid plaques in nearly 30% of patients at 24 weeks and 76% at the end of the study. And in addition to the plaque reduction, donanemab also showed slowing of progression of cognitive and functional decline to a similar magnitude to lecanemab, I think. Depends on the groups that are looked at, but that slowing ranged from about 22 to 35% over 18 months on participants on donanemab. The risks and side effects were similar, although there was less risk of infusion reaction on donanemab, 8.7%, but ARIA was also seen with donanemab. ARIA-E and ARIA-H again, edema and hemorrhage, and ARIA of any form occurred in 37% of patients on donanemab. And again, symptomatic ARIA was only in 6%, so the majority of cases were asymptomatic. But there were patients with symptoms, there were patients who had very serious symptoms, and in fact, there were 3 deaths in the donanemab trial related to ARIA-E. And again, the ARIA risk with donanemab is related to the ApoE4 gene carrier status. So I made this table, but I shouldn't have made it, but it's here. So you can't directly compare these two medicines. They have not been tested in a head-to-head, but there were some similarities between the studies. So I think this could be a bit helpful. Lecanemab was approved in July 2023, donanemab in July 2024. Both are given intravenously. Lecanemab is given every 2 weeks. Donanemab is given every 4 weeks. The treatment duration for lecanemab was given for 18 months in the study, but it may potentially be given longer. And donanemab, according to package insert treatment, may be variable depending treat to amyloid clearance, but probably minimum of 6 months. Slowing of progression with lecanemab, 27%, donanemab 22 to 35%, depending on the groups looked at. The risk of ARIA looks higher in donanemab by group for whether 0, 1, or 2 copies of ApoE4. Risk of ARIA-H looks a bit higher. Risk of infusion reactions looks higher with lecanemab. There were deaths in each study, and the cost per year, again, cannot be directly compared because the treatment course may vary. So what about the patient perspective? Patients are already starting treatment in our clinic and many others across the country. So when I'm talking about these medicines with patients, and treating patients, it is important to realize the infusion reactions can be pretty severe. I think more so than what we saw in trials. We've had patients stop drug because of infusion reactions. We've had patients go to the emergency room after their infusions because they have felt so wiped out. Some patients may not want to be tied to their infusion schedule, extended treatment course, or have a travel burden to come to the infusion centers. Many patients have concerns about side effects, affordability, and regular MRI safety imaging can be unpleasant or unsafe if someone has a pacemaker or something else that prevents them from having MRIs. So if someone cannot have MRIs, they cannot take these medicines. So 2 recent quotes from my clinic. Anything I can do to slow progression, I want that. I want to be there to see my grandkids. And a patient a few weeks later. I just want to live my life right now the best way I can without worry about infusions, side effects, travel limitations. And I think both of these responses are understandable and valid, and it really is an individual decision. So important questions still remain about the use of anti-amyloid antibodies. So this graph is showing some subgroup analysis from the lecanemab study. And essentially with this graph, if we see the black bar to the left of this dashed line, that favors lecanemab in that group. And if it's crossing the dashed line or going to this side, it may not favor lecanemab in that group. And a couple things to note is that the question as to whether or not the treatments are safe and effective for racially diverse populations remains an open question. We can see that there were very few Black participants in the lecanemab study, and there may not have been a benefit in that group. Same with Asian participants. Another question is raised about ApoE4 homozygotes. Should they be treated with lecanemab or other antibodies? What about patients with mixed forms of dementia? So many patients do have more than one form of dementia, not just Alzheimer's as a cause. What is the optimal length of treatment? When can treatment stop? What about patients with multiple medical conditions that were excluded from the trials? Because trials are looking for a very safe, a very healthy population. And what about switching between treatments? Is that possible? Some patients are already asking, can I start with one and then switch to the other? And we really wouldn't recommend that at all. That has not been looked at all in terms of safety. So future directions are very important in this Alzheimer's treatment development. So we need to test these anti-amyloid antibodies in racially diverse populations. We really need real-world efficacy and safety data from patient registries which are being generated. So patients who are being treated with lecanemab and donanemab are being entered into registries who will follow them over the long term. What happens after 18 months? What happens when other medical conditions are at play? Testing of subcutaneous forms is already happening, and so that would be administration not by an IV, but self-administered by a patient or family member as insulin is given or other medications at home, and that would really make things easier. These medicines are being tested in pre-symptomatic populations, so patients without symptoms but who are developing brain amyloid already, and if can treatment early prevent the development of mild cognitive impairment in Alzheimer's dementia. And that's in the AHEAD study and the Trailblazer ALZ 3. These medicines are being looked at in combination therapy in clinical trials already with other mechanisms, including in combination with tau therapies. And testing these medicines in other forms of Alzheimer's is also important, like familial Alzheimer's disease related to presenilin or APP mutations, and potentially treating in patients with Down syndrome and Alzheimer's disease, although we really need to be quite cautious with that because patients with Down syndrome are at higher risk of brain bleeding. And then screening and monitoring of patients with blood markers of amyloid and tau, both for identifying patients who may be eligible for these treatments and then monitoring them over time. So in summary, the future of Alzheimer's treatment is just beginning. But we do know that for patients with early Alzheimer's disease, 2 new anti-amyloid treatments, lecanemab and donanemab, are available and have been shown to reduce amyloid in the brain and slow decline of cognition and daily function. The most concerning risks are brain swelling and bleeding, which is called ARIA, and this risk depends upon ApoE4 genetic status. The treatment course is prolonged and involves frequent intravenous infusions, and patient-guided decision-making is very important. And these discoveries are hopefully just the first among many future treatments that will benefit patients and families with Alzheimer's and other forms of dementia. So thank you very much.
Time for some questions. I see some hands and I see people with microphones running.
Dr. Niam, thank you very much for your
I don't think
presentation.
it's on. Romina?
There's a problem with the diagnosis, and the clinical diagnosis of Alzheimer's disease is very delayed and takes a lot of time. And I know that because my wife has also dementia. Number 2 is that what do we talk about the blood test like the B270 and the ambulance that's been just published at JMA in July. Where is that going to be in the US in okay by next year? Thank you.
I didn't get the first one.
Okay. So the first was a comment about the risk that diagnosis can be delayed, and I'll connect the dots. I think, the relevance to early intervention.
Yes, so indeed, the diagnosis, you're right, can be delayed significantly for a whole variety of reasons. Sometimes patients may not have insight, but often they do. Sometimes there might be access issues to doctors, clinicians, and innumerable other reasons. But I think that if a patient is already in a moderate or severe stage of dementia, it's not clear that these medicines would be of help, and they should not be used in that stage. In terms of the new blood test, we will get to hear a whole talk about that by Dr. Suzanne Schindler later today.
Thank you for that great lecture. I can be loud. So the question is, I felt comparison to saline is a little misrepresented. No one would get saline. Is there a comparison to current medication like Aricept or Memantine, and how would those compare? Because those definitely have less side effects, and if they're equally or even closely similarly beneficial. Thank you.
Very great question. So actually, in these studies, it is true. Participants coming into the study were allowed to continue any of their currently available Alzheimer's medicines. So donepezil, rivastigmine, galantamine, memantine, they were allowed to be on those medicines at the start of study, and then they were randomized. So participants, probably an equal amount in the lecanemab or donanemab group versus placebo were on those medicines. So these medicines have not been directly tested head-to-head just against donepezil or rivastigmine. However, when you try to look at the curves as to whether or not that meaningful difference is different, if you compare, studies from years and years ago of donepezil to now, actually the magnitude of effect is pretty similar to what we can see with donepezil, but the long-term effects, that's what's not really clear, and there's some hope that perhaps clearance could lead to further gains down the road. But in terms of the actual magnitude, it's not very different at all from donepezil. Well, so in terms of those medicines like donepezil, those aren't thought to actually slow progression. They're thought to show a symptomatic benefit and then a curve of decline, which is similar to the rate of people not on medicine. So it's difficult to directly compare. But I will say that in my practice, I look at these medicines as being complementary. I wouldn't choose donepezil versus lecanemab. I usually initially talk about one of the cholinesterase inhibitors, because that is also for people with mild dementia, and then possibly lecanemab or donanemab in addition.
No.
Hi, Kim with Alzheimer's Orange County. Are patients being followed after the conclusion of the trial? And if yes, are they fine? Market decline when they go off the drug?
That's a great question, and it's very complex. And but yes, patients are being followed. Up at OHSU, we were a site for the phase 2 and phase 3 studies, and all those patients were invited to continue on in the studies. But there can be some difficulties. For example, after the phase 3, there was phase 2, there was a long period where the drug was not available. And then with the phase 3, patients are not coming off the drug in the long-term follow-up. They're continuing on the drug. And so we really don't have information yet about the long-term progression, but that is crucially important.
Let's thank Dr. Pierce.
Thank you.
Okay. Dr. Pierce talked about the importance of genetic testing and its role in risk for these new medications. And our next speaker is one of the world's experts on doing that genetic testing and then delivering genetic results back to participants and patients. And that's Professor Scott Roberts, Dr. Scott Roberts from the University of Michigan. Dr. Roberts is a professor of health behavior and health education at the University of Michigan in the School of Public Health. He's a clinical psychologist by training. He also leads the Outreach Recruitment and Engagement Corps for the Michigan Alzheimer's Disease Research Center. He holds a number of other titles at University of Michigan, but of note, he's been a part of, I think, every study to date under the REVEAL moniker. REVEAL was an original study to randomize people with a family history of Alzheimer's disease to learning their apolipoprotein E genotypes and assessing whether that information could be delivered safely. I think we're up to the 5th 5-year iteration of the REVEAL study and the grant supporting it, and he's been involved in each of them. So you won't find many people with more information on this topic than Dr. Roberts. Thanks, Scott.
All right, well, thanks so much, Josh, for the kind introduction, and thanks to you, Megan, the UCI Mind team, for convening such a wonderful meeting. It's great to be here in sunny California. We don't take sun for granted in Michigan, so as Josh talked about, I'm going to be building a lot on what Dr. Pierce was discussing in terms of use of genetic testing. So I'll start with an overview of genetic testing for Alzheimer's writ large, and then towards the end, pick up where Dr. Pierce led off on how does genetic testing relate to these new anti-amyloid therapies that are now on the market. But before I begin, just to mention, even though I'm going to be talking a lot about genetics, I think it's important to note that Alzheimer's is a complex multifactorial condition. So even though I'm going to be focusing on genetics, that's certainly just part of the story. As you can see here, there are a lot of risk and protective factors that are relevant. And just because someone has genetically at elevated risk for the disease does not mean those protective factors aren't relevant, that you shouldn't continue to do the brain healthy behaviors that you probably heard a lot about. But in terms of genetic testing, this has been around for decades related to Alzheimer's disease. You can see this listing here shows that not just for Alzheimer's, but throughout medicine, there's all kinds of different ways genetic testing can be useful. I'll talk some about predictive and susceptibility testing, which means testing asymptomatic individuals to learn more about their risk of a condition. There's, in rare cases in Alzheimer's, it might be used to confirm a diagnosis. There's something called pharmacogenomic testing that you may have heard about, where genetic testing is used to inform decisions about medication use. So I think this is a very valuable tool, not just in the treatment of Alzheimer's, but in medicine writ large. Some of you may know cancer genetics is really a flourishing field as well. This table talks a little bit more about the different types of genetic testing that we do for Alzheimer's particularly. So when I say predictive testing, what I mean is these are tests for rare genetic mutations where if someone carries them, they are almost inevitably going to develop Alzheimer's disease. Whereas susceptibility testing is also testing asymptomatic people, but the meaning of the result is not quite a yes or no answer. It just talks about elevated risk. And then, as I mentioned, pharmacogenetic testing is really focused on informing medication choice. And so these different kinds of tests, they are for different purposes. They're with different populations. They test for different genes, and they're done in different testing contexts. So even though I'm going to be focusing on clinical practice, we do these kinds of tests for these different genes routinely in Alzheimer's research. And I'll talk a little bit about some of these tests are now available direct to consumer as well. So just to kind of represent the different genes linked to Alzheimer's, this is kind of a complicated picture here. But if you look towards the bottom, you'll see there's a few genes listed that I'm not going to focus a lot on, but a lot of what are called genome-wide association studies have identified genetic risk for Alzheimer's that are very modest in nature. So any one of them is not really going to predict a high level of risk for Alzheimer's. But if you put them all together, some of you may have heard of this concept of polygenic risk scores, then maybe they might be more useful in predicting one's chances of developing the disease. You heard from Dr. Pierce about ApoE and the E4 allele. I'll talk more about that. But then also you can see on the upper left, there are some rare mutations known as the, on the APP and presenilin genes that are very highly predictive. And so let me just say a little bit more about those. So these genetic tests are done in the context of what's called familial Alzheimer's, where oftentimes many members of the same family will be affected with the disease. And so this refers to testing for these rare mutations found on chromosomes 1, 14, and 21, that if one possesses them, one will almost inevitably develop Alzheimer's. Fortunately, these are relatively rare, so they account for a very small percentage of all Alzheimer's cases. But if they are present, Alzheimer's can occur much earlier than we typically see it. So we've seen even cases in people in their 30s and 40s developing the disease. And their course of disease, the progression of symptoms is often more rapid in the presence of these mutations. So testing is available for these rare mutations. The reason I have this movie poster up is some of you may have seen the film Still Alice, where Julianne Moore won an Oscar for her portrayal of a woman who did have one of these rare mutations. And in the film, it talks about the drama of should her adult children be tested, because in these kinds of cases, there's a 50-50 chance that the parent would pass that mutation on to the children. So I think that underscores this broader theme of genetic testing, where it may not just yield results for the patient, but also have results that are have implications for adult children or other family members. So, this has been around for a while. Again, it's relatively rare, but I did want to point it out because we do see this kind of genetic testing and counseling done in specialty clinics across the U.S. And then when this is done, we really typically see the involvement of genetic counselors. So I wanted to highlight the importance of this particular healthcare practitioner. So genetic counselors, as some of you may know, are master's level clinicians. They have training both in medical genetics and in patient education and support. And typically, genetic counselors meet pretty intensively with patients and family members and really try to provide pre-test education. So don't just check the box on the medical form to order the test without much deliberation. The idea of the pre-test education session is to really think through, do I really want this information given its meaning, its risks, its implications for family? And then genetic counselors not just provide that pre-test education, but then deliver post-test disclosure of results and provide support and recommendations. So I think genetic counselors are really essential in a lot of genetic testing contexts, but the challenge is they're relatively few in number. So at the University of Michigan, where I am, our program only graduates about 8 to 10 genetic counselors a year. So, and they tend to be concentrated in more kind of urban settings and academic medical centers. So, for example, I live in a neighborhood where there were 2 genetic counselors on a neighboring block, but the entire state of Idaho has 0 genetic counselors, at least at that time. And so they're not spread evenly across the U.S. So we'll come back to that in the future, because when we talk about use of genetic testing in the context of AD treatments, we'll be thinking about, well, who's going to be the practitioner delivering the results? All right, so that's a little bit on familial Alzheimer's, the rare predictive testing that's done. But let's talk more about ApoE and Alzheimer's risk. So Dr. Pierce teed me up nicely for this. ApoE is a plasma protein on chromosome 19. As she mentioned, there are 3 common alleles known as E2, E3, and E4 that are one inherited from each parent. And we've known for decades that E4 is linked to a higher risk of Alzheimer's. But it's important to note that the E4 allele is neither necessary nor sufficient to cause Alzheimer's. So you can have Alzheimer's without having an E4 allele, and you can have the E4 allele and never go on to develop the disease. And to make matters even more complicated, ApoE is also relevant for other health conditions. So the E4 allele is also a modest risk factor for heart disease as well. You can see from this table here, the E4 allele is relatively common in the general population. So if you add up all those genotypes that have an E4 allele in them, you can see that over a quarter of the general population are E4 carriers. Fortunately, the E4-E4 or E4 homozygote combination is relatively rare. Only about 2% of the general population are E4 homozygotes. But when we're talking about, it's very different from those rare mutations I was talking about earlier, where E4 is very common in the general population. What does the E4 allele mean in terms of risk of Alzheimer's? Well, you can see from this table, the general population risk for Alzheimer's over one's lifetime is about 10 to 15%. But if you're an E4 carrier, you can see the risk is about doubled. And if you have 2 copies of E4, you can see it puts one at probably over 50% lifetime risk of the disease. So pretty prominent in terms of Alzheimer's risk. And E4 homozygotes are at much higher risk than that, even for at least presence of those amyloid and tau biomarkers that Dr. Pierce was talking about. If you're an E4 homozygote, it's very likely that at some point in your life, you'll have at least the presence of those in the brain. Although, of course, we know that just mere presence of amyloid plaques, for example, doesn't guarantee that one will have the cognitive decline associated with dementia. So that's just a little bit of background on ApoE genotypes. And so there's been a debate ever since this link was identified back in the 1990s, should we be offering this to people? And so in the medical community, there's been a general skepticism about the utility of ApoE testing, at least for people who do not have any symptoms. So the idea here is, because it's not a definitive yes or no answer, and because there's not a lot we can say that is proven to work in terms of Alzheimer's prevention, the medical field has said we're kind of reticent about offering ApoE testing. And that's because the fear is that there might be some harms associated with ApoE disclosure, whether it's psychological distress from learning elevated risk, maybe social stigma. So to date, you don't see ApoE testing done a lot in clinical contexts with people who do not have a diagnosis of Alzheimer's. But what we found out in our studies is there is a high level of public interest, particularly people who have a family history of the disease are often very interested in learning their ApoE genotype. They feel it might be useful to inform their planning. And a lot of people have this more general sense of, knowledge is power. They feel like if there's information that's relevant to them that's out there, they would like to have it. And interestingly, this kind of testing a few years back became available via the direct-to-consumer genetic testing market. So many of you have probably heard of the company 23andMe, where you just spit in a tube, you send your sample off to them, they give you access to a website, and they tell you about all kinds of personal genetic information, not just for health, but ancestry, et cetera. But as of 2017, the FDA approved 23andMe to include ApoE disclosure as part of this direct-to-consumer genetic testing approach. So controversial among the genetics community, but it's now out there and available, on a widespread basis. So as Josh mentioned, our team at Michigan and many others, Robert Green in Boston, Jason Karlowish at Penn, we've done a series of multi-site trials over the years looking at disclosure of ApoE results, because even though the medical community had these recommendations about not wanting to offer it, there wasn't a lot of data out there on how do people actually respond when given the chance to learn this information. So the REVEAL studies that Josh referred to, we've done for several years now. And so just a few big highlights from these studies I can share here. So fortunately, we had genetic counselors be the ones deliver this information. And I think partly because of that, we had good results in terms of participant understanding of information. People were not assuming an E4 result means they will definitely get the disease, for example. And fortunately, they seemed to adjust well to the information, even if they got bad news. So even people who are learning their E4 status were not responding with clinical depression or anxiety reactions for the most part. So that seemed encouraging. What we did over time was we tried to see, can we deliver this information in a more streamlined fashion, not using this very intensive, time-intensive genetic counseling model with multiple sessions, over an hour of face-to-face time. And so we also found that you can deliver this information in a briefer format. And we found that you can do this, it doesn't have to rely on in-person disclosure. You can do telephone disclosure with similar outcomes in terms of participant understanding and psychological reaction. Interestingly, people did a lot of different behaviors spurred on by their test results. And so I'll talk about some of those now. One thing that people did that we found interesting was many people added some kind of dietary supplement. It wasn't irrational to do this. There's been some observational studies, for example, suggesting, well, maybe vitamin C, vitamin E, ginkgo, those kinds of things might be beneficial. So that was one interesting behavior. But I did want to point out, though, we were a little bit wary of this because I think particularly now we're seeing the growth of this nutraceutical industry with a lot of products on the market that make these general claims about supporting memory functioning, et cetera. And so I do think it is important to keep in mind that these products are not regulated in the same way that medications are. My slogan here is, if it sounds too good to be true, it probably is, but there's a lot of products on the market in this space. Another thing that people did that, again, made some sense was a lot of people who learned they were at elevated risk reported some kind of insurance changes. But I think it's important to keep in mind there are some legal issues here to think about when we think about genetics and insurance. So there is a federal law out there known as GINA. If you've never heard of it, you're not alone. We've done studies that show there's very little public awareness of this legislation, but it's been around since the Bush administration. And GINA prohibits the use of genetic information by health insurers or employers. So it's illegal, for example, for a health insurer to raise your premiums or deny coverage based on your genetic status. This is really important. For example, there's a lot of people who know they're at elevated risk for cancer, for example. It's also illegal for employers to use genetic information against people. So it would be illegal to not hire someone because you're worried that their genetic status might elevate the employer healthcare costs. So I think we see this as a good thing, but in general, GINA does not cover all the potential areas of genetic discrimination one might think of. So GINA does not apply, for example, to long-term care, disability, or life insurance. And that's particularly relevant because long-term care insurance, I think, is the one domain that a lot of people who learn their E4 result might be tempted to want to change, maybe to initiate a new policy or add to an existing policy. And so I think it's important for people to know that GINA does not extend to long-term care insurance. It's not to say that long-term care insurers are going to engage in genetic discrimination, but I think, again, in this theme of informed consent, informed choice, I think it's important that people go into genetic testing knowing about some of these legal issues. Genetic testing, I've been talking about it in direct-to-consumer clinical contexts. Of course, it's done in a lot of research studies as well. It's very routine at Alzheimer's centers across the country to collect all kinds of genetic information. It's so easy to do these days. You just do a buccal swab, little Q-tip in the cheek, and you can get a sample where you can derive DNA from. But typically, most studies, we've done research in this area, are not giving back people's ApoE results in most Alzheimer's research studies. But more sites, I think, are considering this. But interestingly, genetic testing has been done as part of some of the prevention trials that Dr. Pierce was talking about. That is, using genetic testing to see who's eligible for these experimental drug trials in the first place. So some prevention trials have used biomarker testing to identify eligible at-risk populations, whereas others have used genetic testing, both ApoE and for those rare mutations I alluded to earlier. One famous example was the Alzheimer's prevention study done in Colombia, South America. Some people may have heard about this. So unfortunately, this was one of these negative trials, but it was a really fascinating study where they enrolled people who did have one of those rare mutations that was inevitably going to cause the disease, and they were trying to test out an anti-amyloid therapy in these gene-positive individuals in South America. So unfortunately, a couple years ago, the results were announced, and it did not show a positive benefit from the drug. And so I think at that time, there was a lot of discouragement about, are we investing too much in this amyloid hypothesis? But as Dr. Pierce pointed out, we've seen some successes since then in terms of anti-amyloid therapies. So that brings me to this idea of pharmacogenetic testing. So again, as I mentioned earlier, this is this concept that genetic tests, not just in Alzheimer's, but more broadly in medicine, might be useful in thinking about informing medication choice. We see this, for example, in psychiatry. Sometimes these tests are done to try to help select which SSRI to put people on. And I think the reason why this is useful is because these tests can be helpful, particularly in predicting who's going to be basically a poor responder to a medication, unlikely to derive benefit. And as Dr. Pierce alluded to, they can also be helpful in identifying who's going to be more likely to have some of these concerning side effects that most medications carry some risk of. So as was pointed out earlier, ApoE testing is definitely relevant here. So both lecanemab and donanemab have this ARIA side effect that was pointed out earlier, the brain swelling and microbleeds. And as was noted, if you have E4, you're at elevated risk of this ARIA side effect. And particularly if you have two copies of the E4 homozygotes are at even higher risk. And so I think we're seeing kind of an increased attention now to ApoE testing in the context of these newly approved drugs. And this is kind of a complicated table here, but if you look at the upper right, you can see that the E4 homozygotes on the lecanemab study had about 33% chance of having an ARIA-E observed, as opposed to the placebo arm having under 2%. But fortunately, just presence of ARIA-E doesn't necessarily result in symptoms or serious symptoms, so those percentages were much less of a difference between the E4 homozygotes and placebo for ARIA that was resulting in symptoms or more serious adverse events. But given the discrepancy, and we've seen this across multiple trials, and not just for lecanemab, but for donanemab as well, we've seen recommendation for ApoE testing before going on these medications, because it might help inform, for example, should you even go on it in the first place? Or if you do we want to maybe monitor for these ARIA side effects more aggressively via neuroimaging? So, ultimately, I often get asked, well, what should we be telling people? It gets tricky here because, kind of can depend on the context. Again, I alluded to earlier, the medical community has basically said, if you're thinking about ApoE testing for an asymptomatic person with a family history of the disease, the medical community has said, well, we're still pretty wary here. We don't necessarily think the benefits outweigh the risks. I feel personally that's maybe, particularly given some of our results in REVEAL, maybe that's a little too conservative. It can depend maybe on the context as to what's appropriate. But what I do think is that regardless, I do believe in the importance of informed choice and informed consent, because a lot of times people have misconceptions about what genetic tests can and can't tell you. So fortunately, there's some great materials out there. This is a website, genetestornot.org, that is one of these interactive decision aids that kind of walks people through the pros and cons of ApoE testing. There's other great resources, the Alzheimer's Association, the National Institute on Aging, ADIR website, that all have great background information on genetic testing. So I think if you or someone you know are interested in considering this kind of test, those kinds of resources I think might be helpful to look into before trying to take action. And then, but I think that the decision is different, as was mentioned earlier. Lecanemab, these appropriate use recommendations came out last year from a team of luminaries in the field. And as you can see, these appropriate use recommendations for lecanemab said that given ApoE4 carriers increased risk for ARIA, they are routinely recommending ApoE genotyping of all treatment candidates before initiating lecanemab, because it might affect the risk-benefit ratio. So I think this is good to have these recommendations, but for me, it's really interesting to think about, well, how is this really going to play out in clinical practice? As I pointed out earlier, typically it's been genetic counselors to be the ones delivering information. But I think in this context, it's probably not realistic to expect genetic counselors to be the ones giving ApoE information. We're going to be relying on neurologists, geriatricians, even primary care docs. And as you might imagine, this stuff can get pretty complicated pretty quickly. It's very complicated jargon. We're talking about risk statistics. We're talking about genetic information that has implications not just for the patient, but if you're an E4 homozygote, that means any adult child will automatically be an E4 carrier themselves. And so kind of preparing families for this ripple effects of information, I think it's a pretty challenging patient and family education dilemma to think about. And so I think even though I agree with these recommendations, I think we also need to be thinking about how are we going to prepare both patients, family, and the workforce for doing this genetic testing in a safe, effective way so that people understand the meaning of results and the limitations. So in closing, I hope you've seen how there are several different types of genetic testing for Alzheimer's that are possible. None are considered standard of care in asymptomatic groups. We talked some about how ApoE testing is now recommended to inform decision-making for the new anti-amyloid therapies that are on the market now. And I hope I've hinted at some of the challenges and opportunities in disclosure of genetic test results and kind of thinking through some of these broader ethical legal and social considerations that attend not just genetic testing for Alzheimer's, but genetic testing more broadly. So with that, why don't I wrap up, and thanks for your attention, and open it up for questions.
For Dr. Roberts. We need a mic. Coming. There are thousands of people online.
Thank you
listening.
so much. Can you comment on the accuracy of the direct-to-patient test, like the 23andMe, and also the insurance coverage? So what end up being the cost to patient in general? Thank you so much.
Yeah, so the 23andMe test is very accurate in delivering one's true ApoE genotype. So to get FDA approval, they had to demonstrate that they're calling the ApoE4 genotype or accurately for any kind of consumer. But does the ApoE4 test tell you risk of Alzheimer's with certainty? As I pointed that out, no. It's just kind of an important risk factor. So presence of E4 doesn't absolutely predict with certainty one will develop Alzheimer's. But at least the 23andMe, you can feel confident you're getting your right ApoE genotype if you do go that route. In terms of insurance, I think partly because some of these recommendations, I'm not aware that ApoE testing is ever covered by insurance for someone who is asymptomatic and who just wants to learn more about their risk. I think that would be more of an out-of-pocket expense if you're just doing it for susceptibility testing purposes. In terms of being done in the context of treatments for Alzheimer's, actually, I'm not 100% sure myself. Dr. Pierce, do you know if the ApoE test, when it's being done to kind of inform treatment choices for these new drugs, in your clinical experience, has that kind of ApoE testing been? Yeah, she's nodding. I think because, and here's where I think a lot of times insurance companies will go by, these expert recommendations because they're considered standard of care. And so those appropriate use recommendations for lecanemab that I mentioned that came out last year that recommended routine use, that will often lead insurers to cover. So I think the answer is genetic testing can be covered by insurance, but it depends on the indication for the testing.
Scott, there's a question online. Could you comment on genetic testing for frontotemporal dementia?
Yeah, so there are, I think similarly in frontotemporal dementia, there are some rare genes, kind of like we saw with the presenilins and APP, that can be done that do predict, development of frontotemporal dementia. But I think there are also, in these kind of situations, these are very rare mutations, so not necessarily relevant for the entire population of FTD patients, but kind of similar in Huntington's disease is another example where we do see these rare mutations in certain cases are really causative for the development of later dementias and other neurological conditions.
I noticed that you said E4 positivity increased the risk for both Alzheimer's and the ARIA side effect risk. So I guess the people who are most at risk or need it most can't really use the therapies. Is there a hypothesis on why it causes the ARIA side effect?
Yeah, I think we're still learning about the linkages and the mechanisms. So to my knowledge, there's not a definitive answer for sure why this association is there. And I think further research is probably going to hopefully illuminate some of that. But I think it's been kind of this consistent finding across multiple trials, and it's been pronounced enough that I think that it's caused the experts in the field to recommend we think this is worth doing, because it does seem to affect the calculus of the risk-benefit ratio of the medication.
Yeah, there are several hypotheses about why that risk is elevated, and one major contributor is likely to be that people who have E4 are at increased risk to develop a condition called cerebral amyloid angiopathy, which means the amyloid is building up in the blood vessels as well as the parenchyma, and so the active process of pulling the amyloid out when the amyloid is already in the vessel could increase or exacerbate the risk for these side effects as well. I think we are going to move to our 30-minute break. Let's thank Dr. Roberts one more
thank you.
time. Welcome back from the break. I am extremely excited about our next talk and even more so our next speaker because we are very fortunate to have recruited Dr. Leigh Turner to UC Irvine in the last couple of years. Dr. Turner is a professor in the program in public health and the Department of Health, Society, and Behavior. He is our new founded director of the UC Irvine Center for Health Ethics. Dr. Turner is world-renowned in his study of stem cell clinics, and I believe this to be incredibly pertinent to our community and the people we are trying to help through research, because I fear that they are being subjected to what is commonly known as pseudomedicines, often with great risk and at great financial charge. And so it's just a fantastic thing for UC Irvine and for UCI Mind that Dr. Turner was recruited into the Stem Cell Center in the School of Public Health. And I am just so delighted that he has agreed to join us today and talk about some of his work and its relevance to Alzheimer's disease. Dr. Turner?
Okay, I'll get started. So this is my first time attending this conference series, my first time attending an Alzheimer's disease conference. So I feel a little bit coming in from the side. I hope I have something here that's of value to you. So let me just start with a couple of thanks. I want to thank Dr. Grill, not just for the introduction, for the invitation, but also for the support and enthusiasm since before I even arrived here. It's really been a great source of support. I also want to thank Dr. Whitbrack. There's obviously all kinds of logistical details that go into these events, and I'm appreciative for her help and those others who organized this event. So as my title suggests, I'm going to discuss marketing misrepresentations that have been made by businesses selling purported stem cell treatments for Alzheimer's disease and other dementias. There are some real issues with those kinds of claims, advertising claims and medical practices. Unsupported by convincing evidence of safety and efficacy of serious implications for the health, the safety, finances, and well-being of persons with dementia and their loved ones. They also damage efforts to conduct credible clinical trials and harm public understanding of stem cell research in Alzheimer's disease. And I think it's those collective effects that are often missed. We maybe sometimes focus on risks to individuals, but there are public implications as well. Now, there are, beyond stem cells, there are many examples of businesses advertising purported stem cell treatments for dementia without having credible evidence to support their claims. And Dr. Roberts mentioned some in his presentation. UCSF neurologist Joanna Helmuth and colleagues describe such products as pseudomedicine for dementia and brain health. They define pseudomedicine as supplements and medical interventions that exist within the law and are often promoted as scientifically supported treatments, but lack credible efficacy data. They write, practitioners of pseudomedicine often appeal to health concerns, promote individual testimony as established fact, advocate for unproven therapies, and achieve financial gains. Now, I think we could actually have an extended discussion in terms of, is pseudomedicine the most appropriate label? Are there shortcomings with that term? And I think that would be a good conversation to have. I think the larger point that I would want to make is that in general, the conversation is about interventions, procedures, products that are not backed by meaningful evidence of safety and efficacy, aren't backed by convincing evidence. And often there's a profit motive lurking in the background, that it's not just sort of done in an altruistic fashion, there's business interests at play here. Now, my presentation addresses stem cell-related marketing misrepresentations, but many other products and procedures are also advertised as treatments for dementia. A broader analysis would examine the marketing of supposed memory-boosting dietary supplements and smart pills, vitamins, herbal medicines, brain games, clinical protocols, and home remedies. Some of this activity has caught the attention of the FDA and the FTC and regulatory bodies in other countries. Concerns about the cost of such products, possible side effects, and the exploitation of patients and their loved ones date back decades, as do criticisms of therapeutic fads and quack care for neurologic diseases. So if you look at this full slide here, one of the articles dates back to the 1990s. That's the use of unproven interventions article. And then the other one, I'm guessing many of you have never come across, it's therapeutic fads and quack care. It's actually written by UCI Medical School. This goes back to 1983. So in a way, we're kind of continuing a conversation that has gone on for decades, and the interventions may change over time, but the larger set of issues, I think, remain and persist. And 2 decades ago, so I'm going to give a bit of a chronology, a very brief chronology. Journalists and academics documented the emergence of clinics marketing putative stem cell treatments for Alzheimer's disease, ALS, MS, Parkinson's disease, and other illnesses and injuries. So a lot of these clinics, if you went on their websites, you'd find just like dozens of claims in terms of treating diseases and injuries. So it's not just Alzheimer's disease. It's kind of like stem cells as a panacea for almost everything. Early reports described so-called stem cell clinics operating in such countries as China, India, and Mexico. The clinics lacked regulatory approvals and credible evidence to support their claims. And there was a way in which businesses could get away with this. In some jurisdictions, such approvals were not required. Operating in regulatory voids and legal gray zones, clinics made claims that attracted hopeful and sometimes desperate patients. And so in a way, what would happen is businesses would kind of set up in jurisdictions where they could get away with what they were doing. They didn't necessarily need pre-marketing authorization, and so they could kind of sit in these spaces and attract patients from around the world. Now, there's a broader environment in which this commercial activity took place. Clinics selling stem cell treatments emerged in a context of growing scientific and public enthusiasm for the promise of stem cells as potential therapies for a wide range of clinical indications, including neurologic diseases and injuries. And so we have a kind of an interweaving taking place at these businesses, because what would happen is the businesses that were selling these purported stem cell treatments, they would often draw upon scientific research. They'd mix exaggerations, hyperbole, and misrepresentations with references to credible studies. And that's a distinction I'm trying to make in this presentation, that misleading marketing claims are the focus of this talk, this critique, stem cell pseudomedicine, as opposed to credible contributions to stem cell research. And so I'm not trying to lump everything stem cell related into this critique, far from it. And this issue, by the way, of studies being picked up and taken advantage of is something many academic institutions have had to deal with. So here at UC Irvine, for example, leaders at the Stem Cell Center sometimes have to deal with like local clinics that refer to studies here. I was previously at the University of Minnesota. Same thing took place there. A very challenging thing for academic institutions when direct-to-consumer clinics kind of like appropriate and mobilize their research in ways that it's not intended. So a little bit more context and history. An early study of online marketing claims made by such businesses was published in 2008. Timothy Caulfield, and this is a particularly well-known paper in this area. Timothy Caulfield and his research team at the University of Alberta found 19 websites advertising purported stem cell therapies. 16 of them claimed to treat neurologic diseases, including Alzheimer's disease. The 23 affiliated clinics, so you'd have like an online portal, a website, and then sometimes you have more than one clinic associated with that website. They might be in the same country or different countries. That's why there's a discrepancy in the numbers. But the 23 affiliated clinics were based in such countries as Mexico, China, India, the Philippines, Russia, and Thailand. And you can see the list goes on. All identified websites marketed therapeutic benefits. Nearly 3/4 of such websites failed to disclose risks related to stem cell interventions. Subsequent studies have also found such businesses making claims about benefits while describing risks as minimal or simply not mentioning them. I think what's interesting is we really haven't got that far into the conference, and we've already heard quite a bit about risks. And I think that's, you need that for a frank, meaningful conversation about interventions. What are the possible benefits? What are the possible risks? Do the possible benefits outweigh the risk? You need kind of like honest, forthright disclosure of information to even begin to have that analysis, even begin to have that discussion. And so if you have a business that touts benefits and trivializes or doesn't disclose risks right out of the gate, you've got a problem, a major problem. Now, there's actually a missing piece. A lot of the articles from this era, sort of 2004 to 2010, they'd use the language of stem cell tourism. And there was an assumption that this involved, travel to international clinics. There was some truth to that, but it missed some commercial activity. And I'm just going to mention that. So most early analyses of businesses engaged in online direct-to-consumer advertising of stem cell treatments, they overlooked US-based stem cell marketing schemes, even though they too emerged in the early 2000s. And so this is kind of like the little known part of the story. In 2006, for example, two US women, a doctor of osteopathy specializing in the treatment of Lyme disease and an associate, were convicted of fraud for a stem cell scam that began in 2002. The women targeted patients by marketing stem cell therapy as a treatment for ALS. Victims were told they had to pay $35,000 to participate in what was presented to them as an FDA-approved study of stem cell treatments for ALS. And so I'm mentioning this just to say, what sometimes people will say, like, well, just, give people information, let them know about red flags to look for and kind of, caveat emptor, buyer beware, let people navigate this marketplace. It is very difficult to actually identify problematic claims because if someone tells you that you're, entering into an FDA-cleared, FDA-approved clinical trial, that may seem plausible and persuasive, and it may take quite a bit to find out that's not the case. This case, if you're interested, was prosecuted by Chris Christie. So it's not as though this is ancient history where all the players have gone away. In a second criminal case, this is also not one that's terribly well known, the founders of US-based Biomark, a businessman and the two founders were a businessman and a former model. They were accused of conducting another stem cell scam that began in 2002, a busy year for stem cell scams in the US. The defendants were charged with 51 criminal counts for allegedly defrauding 25 individuals suffering from ALS, MS, Parkinson's disease, muscular dystrophy, and other diseases. But before they could be tried, the couple fled the US as fugitives. And so that's one, the criminal indictment is online. If you're interested, you can actually learn quite a bit about how this scam operated. In terms of the information that's online, something about the victims, the 25 victims, how they were drawn in. And in this case, there's personal tie to where I used to work. This business did refer to studies that had been conducted at the University of Minnesota to justify what they were advertising. And when that PI was contacted, they said, this is complete misrepresentation, this is fabrication, this in no way, accurately characterizes our research. Now, you might think that given this, sort of robust, strong response, that nothing would really emerge in the United States. But in fact, despite these early criminal cases, US businesses marketing purported stem cell treatments proliferated. By 2016, UC Davis stem cell biologists and I found 351 US businesses selling purported stem cell therapies, with 27 of them advertising stem cell treatments for Alzheimer's disease. None of these interventions, just to be clear, were FDA approved for treating patients with Alzheimer's disease or other dementias. None had first been tested for safety and efficacy in FDA cleared and institutional review board approved clinical trials. So this is just, putting out a shingle on the internet, making claims about treating diseases and injuries, not doing basic research first, not doing preclinical research, not doing phase clinical trials. It's just kind of going online and making compelling advertising claims. The marketplace expanded dramatically. By 2021, this marketplace had expanded to 1,480 US businesses, operating close to 3,000 clinics. It's actually so large that my research team and I mean, it's become very challenging to study, because when you're trying to go grind along business by business and code them and analyze them, there's so many. It's like the project that never reaches a finish line. It just keeps going. So very large marketplace. 134 of these businesses claim to treat neurologic diseases, with 37 promoting stem cell treatments for Alzheimer's disease. So in terms of this direct-to-consumer stem cell marketplace in the United States, neurologic diseases and Alzheimer's disease is a comparatively small part of it. But it's still, these are still businesses that we don't know how many patients they attract, but it may be quite a number of individuals. I was involved in a class action lawsuit against a business in Florida, and in a 3-year period, it marketed stem cell treatments for COPD, other lung diseases, and it had over 1,000 patients go to just one of its clinics. Now, drawing from an ongoing global study, Dr. Grill and I have talked for quite some time about doing a project together, and I'm just describing, kind of the beginnings of that project. I'm going to try not getting into the weeds. It's very easy to go down that path, but I'm going to try and just give you a broader sense of what's coming out of this work. Looking not just the United States, but at a global marketplace, it's been possible so far to find 122 businesses currently advertising stem cell or stem cell derived exosome treatments for Alzheimer's disease, other dementias, and memory loss. Now, 30 of these businesses are US based, 92 of them are located outside the United States. And I'm expecting these numbers to bump up a little bit because you find leads, you find businesses to analyze, and then you have to kind of go through that. So I know it's going to go a bit past this, but this is where it's at right now. The US, Mexico, and India are 3 of the leading hotspots for such businesses in their clinics, but they can also be found in such countries as Russia, Thailand, and Ukraine. And what I want to mention here, and give you a bit of insight into, is not so much the minutiae of, what kind of cells do they use and details about mode of administration. I think that's interesting. I care it's collected this data, but I just want to give you a sense of some of the sales pitches they use. And I think that if you're a patient or the loved one of a patient, these may be quite compelling. I mean, they're effective. Here's a few examples. So I have not provided screenshots of the businesses. I've redacted the company names. There are pros and cons in terms of name and shame and not disclosing that, but I've decided to redact, and I can explain why later if you're interested. Not wanting to get sued and not wanting the conference organizers to get sued is a factor. So one business uses the language of miracles in its pitch. The company states, intrathecal injection of mesenchymal stem cells has emerged as a miraculous therapy for early onset dementia recovery. The results from the early onset dementia stem cell treatment can last for years or even indefinitely. This is because the stem cells are creating real neurogenesis, which means new neurons. I think we can even begin to see here kind of like a blurring of advertising and hype, but also some of the goals of credible stem cell research, which I think can make this so challenging for many individuals to kind of like assess these claims. Another business promotes stem cell cures for Alzheimer's disease, so outright cures. The company states, Alzheimer is very effectively curable with the help of stem cell therapy treatment for Alzheimer's disease in India. So this is obviously one of a number of clinics based in India. Can stem cells cure Alzheimer's disease? Absolutely, yes. The patient will lead a normal life again. So there's a narrative power to these kinds of claims. If you have been diagnosed and you're going online, this is an appealing and compelling pitch. A third business claims stem cells can regenerate new nerves, brain cells, and in some cases reverse 100% the effects of fibromyalgia, Alzheimer's, and Parkinson's, to mention just a few. And again, remember, it's a lot of diseases that these businesses claim to treat. Think of stem cells as hitting the reset button for your body, allowing your healthy cells to be loaded back into the system and flushing out the errors. Again, I think this is actually, as a sales pitch, it's quite powerful. I mean, it's vivid, it's brief, it kind of gives you insight into what they're claiming to do. Actually doing this would be an incredibly complicated, technical feat with many challenges. A fourth company, this is a somewhat related pitch, but I'll mention it. A fourth company claims these cells are akin to having a personalized fire rescue and repair crew that naturally resides in your body. They wait quietly for a sign of trouble, inflammation, and then home to that signal to do their jobs. By harnessing the power of your own biology, the company has its name, represents a minimally invasive option for patients with neurodegenerative conditions such as Parkinson's, dementia, and Alzheimer's disease, or after a stroke or traumatic brain injury. And again, it's usually like one stem cell product, and it's like all these different diseases and injuries they're claiming to treat. Other businesses claim the stem cell products they sell are better than existing medications for Alzheimer's disease. One business states, stem cell therapy helps to improve the symptoms of Alzheimer's disease. Through this treatment, the patient's memory function gets improved, neurons in the brain get regenerated, overall functional recovery gets improved, and the damaged cells get replaced with healthy cells. This business provides stem cell treatment that has no side effects and is a better method of treatment than the use of drugs. And so, when you hear a presentation and there is a kind of an honest discussion of risks associated with drugs, this is the way in which that information about risks can actually get manipulated and mobilized, that someone can come along and say, well, we're taking cells, it's from your body, or it's from, birth tissue derived products. There's no risks. And look at all the other products that are out there on the market. They've got risks. And so better to come to our business. Another business uses the rhetoric of personalized medicine to sell purported stem cell therapies for neurologic diseases and injuries. So this almost sounds like a pitch coming out of UC Irvine and a lot of other places. Personalized medicine, precision medicine, same tropes. It claims our neurological disorders treatment program encompasses cutting-edge adult stem cell therapies designed to address a spectrum of neurological conditions, including autism, ALS, Alzheimer's disease, MS, Parkinson's disease, and traumatic brain injuries. Like same thing again, right? All these diseases and injuries. We aim to halt or reverse the progression of these disorders. Each patient receives a personalized treatment plan tailored to their specific condition, which I think sounds rather, personalized and reassuring. So that's just to give you a sense of some of the, I've called them pitches, the narrative frames, the way in which they're packaging what they're advertising to prospective clients. We'll talk a bit about some problems with this before wrapping. None of the businesses using these sales pitches first published peer-reviewed research providing convincing evidence of the safety and efficacy of the stem cell interventions they sell. Clinical trials testing investigational stem cell products for persons with Alzheimer's disease are underway, but there's a challenging journey of clinical translation from conducting such studies to having legitimate stem cell treatments for Alzheimer's disease or other dementias. At present, there are no stem cell miracles or cures for Alzheimer's disease, no stem cell elixirs or stem cell soups, as they're sometimes called, that magically reset and repair the human body. So I provided a brief excerpt from the Alzheimer's Association in terms of their analysis of current state of stem cell research. And then the International Society for Stem Cell Research has a quite helpful overview of the process of clinical translation that goes into kind of bringing investigational stem cell products from basic research to the commercialized context. And it's a challenging journey where lots of once promising interventions, don't make it past phase 2, phase 3 studies. So a few ethical issues. One major ethical issue with businesses selling stem cells as treatments or cures is the deceptive nature of such representations and omissions. And it's not just what's said, it's also what's not disclosed. The omissions are important. Patients cannot make informed decisions and provide informed consent when they are provided unsubstantiated claims about benefits while risks are minimized or not mentioned. And if you're interested in looking at kind of a juxtaposition between the marketing claims and kind of practical guidance in terms of ethical standards, ISSCR has some guidance in terms of, what informed consent standards ought to be met when stem cell interventions are provided outside clinical trials. Businesses provide inaccurate information on company websites and social media platforms, often target patients with life-threatening or life-altering diseases and injuries. Taking advantage of the hope, desperation, and suffering of ill patients and their loved ones, businesses exploit vulnerable persons. My research collaborator and co-author, Dr. Jeremy Snyder, explores this phenomenon if you're interested in much greater detail in his recently published book, Exploiting Hope. So I just put it here in case you're interested in learning more about kind of the exploitation side of this industry. Knowing many businesses cannot afford such interventions, some businesses provide crowdfunding tips. It's not as though everyone has thousands or tens of thousands of dollars. A lot of people they're looking for help to cover these costs. Crowdfunding further spreads businesses' misleading claims as campaigners plead for financial assistance. When conducting a study of crowdfunding for individuals seeking stem cell treatments for neurologic diseases, Dr. Snyder and I found more than 1,000 such campaigns, and many of them mix promotional claims made by the businesses, factoids, various assertions with very wrenching narratives of hope. In some cases, patients have sued businesses for fraud after concluding they were scammed. For example, former clients with Alzheimer's disease, many of them here in California, were among the members of a class action lawsuit settled by a California clinic that marketed stem cell treatments for Alzheimer's disease, MS, and other indications. In many cases, however, when businesses use deceptive marketing claims, patients lose what they paid and suffer financial harm. Losses can range from thousands to tens of thousands of dollars. And in this case, the business, after settling, it also declared bankruptcy. So I don't actually know if individuals ended up receiving any kind of portion of this payment. That's a common move, is to just kind of declare bankruptcy and go belly up. Some patients suffer harms far more severe than financial losses. In one case, an Australian woman with advanced dementia visited a clinic in Sydney, Australia. She died several hours after undergoing liposuction, meant to collect fat-derived stem cells for a planned autologous stem cell treatment. Clinicians report additional cases of patients suffering complications after undergoing unproven stem cell interventions for neurologic diseases and injuries. One research team recommends creating a registry to track such complications, noting that most such cases go undocumented. And in the survey they conducted, they reported treating such the surveyed clinicians reported treating such complications as stroke, sepsis, seizures, meningitis, and a spinal cord tumor. So this is not just money as losses. Businesses selling unproven and unapproved stem cell interventions likely divert some individuals from participating in clinical trials. Miracles, cures, and treatments are more compelling pitches than being invited to enroll in trials where risks are disclosed in detail. Stem cells are framed as investigational interventions with no assurance of benefits, and there's a possibility participants will be randomized to study arms in which stem cells are not administered. There's also the loss of scientific knowledge that comes from businesses selling purported stem cell treatments without generating and sharing peer-reviewed data that might advance research and benefit patients. So this is where I'm suggesting harms aren't just individual, they're actually collective in nature. Finally, marketing misrepresentations harm public understanding by circulating misinformation and generating confusion about the current state of stem cell research and evidence-based treatments for Alzheimer's disease and other dementias. As investigational interventions tested in well-designed, carefully conducted, and rigorously overseen clinical trials, stem cells are not pseudomedicine. That therapeutic potential doesn't make it justifiable to advertise stem cell treatments now as safe and effective treatments capable of halting and reversing the progression of dementia. So I think just having a public conversation about stem cells and Alzheimer's is difficult when you've got so much information pouring out, not just from company websites, but on, Facebook, YouTube, what used to be called Twitter, and other kinds of platforms. And last point that I'll make, deceptive marketing of stem cells and other unproven and unapproven interventions for patients with Alzheimer's disease and other dementias is not, as I tried to emphasize, confined to so-called stem cell tourism to international clinics. The phenomenon also occurs here in Orange County, as a recent guilty plea reveals. Such misrepresentations leave injured and scam patients in their wake. They also generate public uncertainty and confusion about how to distinguish stem cell scams from the testing and investigational stem cell products, incredible clinical studies, and evidence-based therapies. So this was released, I think, just like over a week ago at someone in the local area who was CEO of two businesses that sold contaminated products that went to clinics across the country. They've been covered in CDC reports because they suffered infections, serious complications, and they just pleaded guilty to one count. As I said, a local side to this, really global story. So, okay. Okay. Is there time for a few questions?
We will make time for a question or two.
I've been a chiropractor for over 4 decades. So I did sports medicine, and I'm approached all the time by people who go, what do you think of stem cells for joints? And I'm also marketed to by these people. I tell them, get out of my office, because you are a kid and then you become an adult and then you age appropriately. So to add a stem cell from either someone else or you is very dangerous. My problem is I am a specialist with seniors with HMO, and the MDs don't know what their patients are actually doing. They're online for supplements, which right now we don't have dehydrated stem cells you can put in your coffee. It's coming. I'm sure somewhere somebody's going to market this. How can a doctor, medical, chiropractic, acupuncture, whatever, protect the patient if we can't integrate with the whole system for this kind of marketing for something in the mind? Because the nervous system controls everything, and you start injecting somebody else's DNA. We just read about DNA, right? How do we protect the patient? Because they're going to do what they do because they're frightened. And they don't want to believe what their primary care says, what their neurologist says, and they're victims of hope. So what advice can you give the medical profession of all practitioners how to have that conversation and scare the heck out of them. Not that they're going to die of Alzheimer's, but they're going to die from the complications.
Mhm. Yeah, no, thank you for your question. So in the work my team has done, we tried to look at some of these businesses in the States. And as you mentioned, I mean, you can find chiropractors offices marketing stem cell treatments in the US. Yeah, you can find podiatrists, naturopaths, health spas. It's this very messy marketplace with an awful lot of players. I mean, I think one piece of what you're getting at is, how do you help people make meaningful choices? How do you help them make good decisions? There are certainly some good resources out there. I mean, I mentioned International Society for Stem Cell Research, for example. They've done a good job of making information about, what you should look for, what red flags to look for when businesses are making problematic claims. The International Society for Cell and Gene Therapy has taken a similar approach. There are some problems with what they do because they often kind of come up with a report. It's a PDF document, it's there online. That doesn't really compete very well with flashy YouTube videos and online testimonies. So there's kind of an information asymmetry, the sober academic approach, and then these really compelling claims. There's also maybe a gap where you have, stem cell societies taking this stem cell scientists, stem cell researchers taking this quite seriously. But many other professional societies not necessarily doing as good of a job in terms of, providing information, providing resources for that particular group of clinicians. So I think there are, many different areas of clinical practice that would probably benefit from having a, much greater focus, much greater attention to having conversations around unproven and unapproved interventions, whether it's stem cells or exosomes or lots of other things that are circulating out there in the marketplace. In the end, would that kind of information, persuade every person coming into a clinician's office? I'm sure it wouldn't, but I think at least some, improved information resources would be a small piece of this. Beyond patients, beyond resources, I mean, I think there needs to be, this is going beyond your question, but I mean, I think there's an important regulatory side to this, that we can't just talk about individual patients and their loved ones and what they need to learn. We need regulatory bodies doing their job and having the resources to do their job so that when a business is making egregious claims, someone is coming in to do an investigation and take steps to stop that from continuing.
Great. I think we're going to actually need to move on, but I think it was a fantastic talk. Thank you so much, Dr. Fern. So I am also very excited about our next talk, and in particular that we are welcoming a new faculty member to UC Irvine to give the next talk as well, and that's Dr. Lydia Ann. Dr. Ann is a new assistant professor in the Department of Psychiatry and Health Behavior in the UCI School of Medicine. She did her medical training at UCLA, her residency at UCI, her fellowship in geriatric psychiatry back at UCLA and the West LA VA. We're beyond thrilled that she has joined us. She brings incredible expertise and knowledge, and she is going to talk about another major advance that we have seen with a first ever drug approved for the treatment of agitation in Alzheimer's dementia and other advances in that area. So, Dr. Ann?
Right. Wow. Thank you for that warm introduction, Dr. Grill. I am thrilled to be here. Right here. Okay, perfect. This is also my first time at this conference, so I'm just so inspired and awed that there's so many people, such a strong support for this important cause, that everyone's rallying together. So just honored to be here and happy to be part of this day. Thanks for having me. So thank you. I'm very excited to talk today about the first approved treatment for agitation and where we go from here. A quick introduction. Over 57 million people across the world have dementia, and that prevalence is anticipated to double every 20 years. Cognitive, behavioral, and psychological symptoms of dementia, also known as BPSD, include anxiety, depression, psychotic symptoms like delusions and hallucinations, as well as behavioral issues. And these can be broad: agitation, aggression, disinhibition, hypersexuality, wandering behaviors, sleep issues, motor issues. All of this fall under this wide umbrella. This is very common. As high as 70% of people with Alzheimer's also have BPSD symptoms. And although cognition is kind of the defining feature of Alzheimer's, this BPSD set of symptoms is actually the most complex, stressful, and most costly aspects of care, often leading to poor outcomes like increased morbidity, mortality, hospitalizations, and nursing home placements. So a very big problem for a lot of people. Now, how do we address this? So first things first, non-pharmacologic options should always be the first line over drug treatments. So we first want to address any underlying causes of these BPSD symptoms like pain, delirium, or constipation. Once these are addressed, there's a variety of different therapeutic techniques. For example, music therapy, aromatherapy, dance therapy, even animal-based interventions. It's a wide variety. So a systematic review looked at a bunch of these different modalities and found that training paid caregivers in communication, patient-centered care skills, or dementia care mapping can actually decrease the rate of agitation by 30% without any medication modifications. The goal is to communicate with people with dementia, to try to understand and fulfill their wishes and their needs. They also found that music therapy, all these sensory-based interventions, can really go far to decrease emergent agitation and decrease symptomatic agitation in care homes. Things that need more data, group activities, and how do we bring these activities to the patient's own home? Things that are not effective, therapeutic touch. All right, so where did we come from? Now for this rest of this talk, I'm going to focus more on the pharmacologic options. I'll be using the term first generation or conventional antipsychotic, which is on the right-hand side, and that versus atypical or second generation antipsychotic. The difference is largely that serotonin modulation that's circled in red. So a peek into our history. Conventional antipsychotics have long been used to treat neuropsychiatric symptoms in dementia, and their overuse in nursing homes led to increased federal oversight in 1987. In the 1990s, we see a shift from conventional to atypical antipsychotics, largely due to a lower risk of extrapyramidal symptoms and tardive dyskinesia. That mechanism is attributed to the serotonin receptor that's marked in red over there. In 2001, more than 70% of atypical antipsychotic prescriptions were used off-label. So not for schizophrenia, not for bipolar disorder, but for dementia. 70% of prescriptions. That's crazy, that's a lot. But in 2002, concerns of metabolic risks of second-generation antipsychotics like strokes and diabetes began to arise, such that in 2005, the FDA issues a black box warning stating treatment of behavioral disorders in elderly patients with dementia with atypical antipsychotic medications is associated with increased mortality. In 2008, they expanded this black box warning to conventional antipsychotics as well. This is a graphical representation of our history with antipsychotics. This second line here is where that black box warning in 2005 came out. But what we can see is that the use of both conventional and atypical antipsychotics for patients with dementia already began declining well before that 2005 black box warning, what we call the no warning period. And this is despite an increase in the absolute number of patients diagnosed with dementia. We also see a shift in prescriptions from conventional to more atypical antipsychotics. This is likely due to an increased emergence of early consensus statements, letters, health system communications, pharmaceutical company actions, and articles attributing strokes and metabolic risk with antipsychotics. This all began to emerge around 2002, such that in 2003, the Office of Inspector General issued a compliance guideline restricting the ability of pharmaceutical companies from marketing off-label indications of medications. So this likely contributed to the decline in antipsychotic prescribing into and throughout that early warning period as well. So after 2005, We see a significant decline in the use of atypical antipsychotics, likely due to that black box warning, such that by 2007, the overall use of antipsychotics declined and leveled off to about 12%. By 2000, now, interestingly enough, medications like cholinesterase inhibitors, memantines, they did not increase during that period, so the prescriptions weren't necessarily shifting there. All right, what happened next? Well, the prevalence of antipsychotic drug use in dementia continued to decrease, down from 31% in 2000 to 20% in 2012. There was also a corresponding decrease in anxiolytics, hypnotics, and sedatives, but actually a 10% increase over those 12 years in antidepressant use. So likely the medication was shifting towards that antidepressant category. Now, interestingly enough, the patients who were prescribed antipsychotics were on heftier doses. So during this time, that defined daily dose almost doubled from 22 to 42. So very hefty doses. All right, now we can't talk about antipsychotics without talking about the CATIE-AD trial, which evaluated whether or not antipsychotics were effective for reducing agitation in patients with dementia. They found that antipsychotics may be effective for particular symptoms of dementia, such as anger, aggression, and paranoia, but unfortunately did not appear to improve overall functioning, care needs, or quality of life. Now, what about other agents? Well, the CITAD study in 2014 looked at whether or not citalopram separated from placebo in improving agitation in dementia, as measured by the neurobehavioral rating subscale. Here we see that by week 6, we see a statistically significant separation in the citalopram 30 milligram arm compared to placebo. All right, now there's also other trials investigating the use of dextromethorphan in patients with agitation related to dementia. And if that medication sounds familiar to you, you're right. This is commonly used as a cough suppressant, interestingly. And when you combine it with quinidine, it's marketed as Nuedexta. When you combine it with bupropion, it's marketed as Alevity. And there's 2 studies, the ADVANCE-1 and the ACCORD trial, both showed a reduction in the Cohen's-Mansfield Agitation Index, which we're going to go through a little bit later. Both showed a reduction in that scale compared to placebo or bupropion alone. This was anywhere from a 6.7 to a 15-point improvement. There was also a 3.6-fold lower risk of relapse and a delay in time to agitation relapse. So pretty promising. Stay tuned because the ADVANCE-2 trials are anticipated to conclude in June 2025. Things that are less promising are agents like masopuritine and prazosin. Originally, it was thought that masopuritine could alleviate BPSD symptoms because it had serotonin, glutamate, cholinergic mediation, so all of these potentials. But unfortunately, this didn't pan out in clinical trials. Similarly, prazosin was promising in original neurobiologic studies, but in the PEACE-AD trial, it did not show an improvement over placebo for agitation over those 12 weeks. So all of this has led to some fairly complex prescribing pathways, such as this one that you see here. There's multiple arms, multiple risks and benefits to consider, and no clear direction, which means that providers are left to balance limited efficacy and adverse effects of medications against a lack of truly effective alternatives and pretty severe BPSD symptoms, and of course, family desperation. So all these decades of off-label antipsychotic use, throughout all this time, that golden goose of FDA approval remained elusive until May 11th, 2023, the FDA granted Otsuka Pharmaceutical Company and Lundbeck Incorporated supplemental approval of brexpiprazole, which they marketed as Zexulti, for treating agitation linked with Alzheimer's disease. This application was granted Fast Track designation and was based on 2 12-week randomized clinical trials, the first of which focused on 1 to 2 milligrams fixed dose of Zexulti, and the second one was a flexible dose of 0.5 to 2 milligrams. Their primary endpoint was the Cohen's-Mansfield Agitation Index, which we'll go over in a little bit, and how much that changed compared to baseline over a 12-week period of time. Okay, let's back up a little bit. What is brexpiprazole? What is Rexulti? The chemical compound is up there. This is an atypical antipsychotic that's already FDA approved for treatment of schizophrenia and also used adjunctively with antidepressants for major depressive disorder. It's a partial agonist at the 5-HT1A and D2 receptors and an antagonist at the 5-HT2A receptors. It's a little bit special in that partial agonism property compared to other antipsychotics. All right, now the primary endpoint for their studies was this Cohen's-Mansfield Agitation Inventory, a scale that asked caregivers to assess the weekly frequency of 29 behaviors from a score of 1 to 7. So that gave you a range of anywhere from 21 to 203. Now, what's considered clinically meaningful is a change of 17 points. 17 points. So we'll keep that number in mind for later. The first study showed that brexpiprazole 2 milligrams did have a statistically significant improvement in the CMAI score compared to baseline at week 12, but the 1 milligram did not. The second study, they found no difference in placebo 0.5 to 2 milligram flexible dosing compared to placebo, unfortunately. However, in post hoc analysis, they found that the subgroup of patients that were titrated to that maximum 2 milligram dose did actually demonstrate a statistically significant improvement in the CMAI score compared to placebo. This was about a 5.3, 5.4 point difference. From a safety perspective, the 0.5 to 1 milligram group of brexpiprazole actually had very similar rates of adverse effects compared to placebo, but those who were on the 2 milligram dose did have a higher rate of adverse effects. Those were most commonly things like headache, dizziness, urinary tract infections, neuropharyngitis, and sleep disturbances. The more serious ones are like seizures. Now, brexpiprazole, like other antipsychotics, will still retain that black box warning that elderly patients with dementia-related psychosis treated with an antipsychotic medication did have an increased risk of death. However, unlike other second-generation antipsychotic medications, brexpiprazole demonstrated a 4-fold increase in mortality. 4-fold. Now that's compared to around 1.6 to 1.7-fold increase for other second-generation antipsychotics. As you can imagine, this FDA approval created quite a stir in the community. Several advocacy groups like Alliance for Aging Research, leaders engaged in against Alzheimer's disease and Us Against Alzheimer's all urged the FDA to approve brexpiprazole. But there is some concern by the community that there might have been commercial interest that was fueling this urge. For example, LEAD is a coalition of more than 200 organizations, including Otsuka, and Alliance for Aging Research does receive funding from Otsuka and includes Otsuka representatives amongst its 17 partners. Now, clinician response is likely to vary based on their pre-existing views about prescribing antipsychotics in this population. Those who are opposed to it probably won't start because of this FDA approval, but those who are on the fence or already prescribing antipsychotics for this population may be more inclined to prescribe brexpiprazole. There's also some concern that this FDA approval could undermine a long-standing message from the Center of Medicare and Medicaid Services to minimize the use of antipsychotics in this population. All right, now on to some controversial points. Brexpiprazole is very expensive. First and foremost, the cost makes it very hard to access for a lot of patients. It's $1,400 a month. Yeah, a lot. We also talked about this already, but there is that concern that the FDA's decision to give them approval may reverse years and years of efforts by the CMS to reduce the widespread off-label use of antipsychotics in residential care homes. And another interesting point is that brexpiprazole still retains that black box warning, but it shows a 4-fold increase in mortality compared to our other options, which shows a 1.6 or 1.7-fold increase. So pretty significant increase. And another point is that the study 1 and the study 2 only showed a 5.4 point maximum improvement in the CMAI scale. But as we talked about, what is clinically significant is considered a 17-point change or more. So is that important? Not sure. Now, this is also very interesting. The results of study 213 were quite unusual. Brexpiprazole produced no benefit at any of the US sites, which enrolled almost half of the patients involved in this study. That 5.3 point difference that we talked about is a result of a larger 9-point difference seen across 5 Eastern European countries and Spain. There was no benefit at any of the US sites. Now, this 9-point difference is also an outlier. It's a very strange number because other phase 3 trials and earlier trials involving other antipsychotics only showed about a 2.3 to 4.4 point change. So 9 points is very large. Some people expressed disappointment in this additional indication based on weak data and wonder if the FDA is lowering their standards. Now, for a lot in the community, this move by the FDA has left more questions than answers. Was there evidence that brexpiprazole provided clinically meaningful benefit? Was there evidence that its risk-benefit profile was superior to other antipsychotics that are currently prescribed off-label? And was that lack of effect at that US sites? What does that mean? Is that important? The FDA responded with their bottom line. That minimally clinically important difference is not easy to determine, and it's hard to tell if brexpiprazole crossed the threshold. And they advocated that the extreme variability in presentation and severity of agitation in patients with dementia calls for the need for individualized risk-benefit evaluation with patients, doctors, and their loved ones. All right, now, despite this confusing history of brexpiprazole, the future is a bright one. There are multiple promising pharmacological targets for BPSD, including serotonin, dopamine, and norepinephrine receptors, as well as muscarinic, glutamatergic, even cannabinoid pathways. So a lot of new studies, a lot of more activity will be shown. So there are some current active studies targeting serotonin pathways. These include several promising serotonin receptors like 5-HT2A, 5-HT1A, and also the serotonin receptor transporter. And in particular, one medication like pimavanserin, which is depicted on that border over there, is a promising candidate. It's already being used for Parkinson's disease-related psychosis like delusions and hallucinations, and potentially has a role in Alzheimer's disease as well. All right, now more ongoing research in these areas. The escitalopram is being investigated in the SCTAD study. There's also the cannabinoid CB1 receptor. There's the glutamate receptor and NMDA receptors, the muscarinic M1 and M4 receptors, as well as 5-HT6 receptors and the dopamine D1 and D3 receptors. These all have kind of different mechanisms of potentially contributing to psychosis and aggression. So I think the future is going to be very interesting once these results come out. All right, thank you for joining me today. We covered a lot of ground, including the history of BPSD treatment, the safety and efficacy of the recently approved treatment, and also future potential targets. These are the sources that I used. And this is my personal inspiration for going to geriatric psychiatry, my late grandparents. Thank you so much for your attention. I'm happy to take any questions.
Time for a question or two, and I already see hands.
I just had a 52-year-old man come in. He's on antipsychotics for 20 years. He was not psychotic. He woke up one night, couldn't breathe. He has a really thick neck, and I thought he might have had a heart attack 20 years ago, right? Or sleep apnea or something.
Sleep apnea, yeah.
His primary care doctor said, "Oh, I have anxiety. It makes me wake up in the middle of the night." Off-label use of an anti-serotonin, 20 years. His insurance, as we just had an insurance question, Aetna refused to cover it because he didn't have a psychotic diagnosis or dementia diagnosis. He's sitting in my office with splitting headache. And I said, you need to have this reevaluated. He goes, well, I did. So my HMO doctor said, well, if you want this drug so badly, you'll have to say that you have dementia or that you have anxiety or bipolar or something. So now he had to sign paperwork that said he had a mental illness. And that with long-term care insurance, he's never going to get it. And I said, don't you dare get off this drug without medical supervision. It could kill you getting off of it. But he was so quiet. It worked for agitation. For anybody else who comes in with a headache, neck pain, they're moving around like, oh, he was just sitting there. And he's only 52. And he's driving on this drug, and it's off-label use. His doctor told him it was off-label, and then he retired 2 years later. But 2 other doctors put him on it until Aetna said, no more. We don't cover it without a psychiatric or agitation diagnosis. How can we protect the public at that young age? And what is his chance of developing dementia if he's not moving around anymore?
Yeah.
Because at 52, he may not make it to 65 or 70. But will that increase his chances of dementia in the future, having been on that drug for 20 years for off-label use?
So yeah, you bring up a fantastic point. First and foremost, we always want to address any sort of underlying cause, and it's often things like pain, things like medical issues. We want to do a thorough investigation of that before starting any of, exactly, yeah. Exactly, yeah. So that's a fantastic point. So we always want to address those first. Whether or not there is a further risk, I think more studies need to be done on the contribution of antipsychotics to dementia. There is some evidence that potentially like cholinergic medications, like a lot of these antipsychotics, can contribute. So you're right in that this polypharmacy is very likely to make it harder for his cognition to function, harder for his quality of life as well. Yeah, it's a very good point. Yeah.
Lydia, there's a question online about whether you'd be willing to comment on CBD use and its use for agitation for dementia.
That's a good question. I have to say, I don't personally have a whole lot of experience on CBD use, but I do hear a lot of anecdotal experience from my patients about CBD use. So I think that this is an area where the research is very hot. I think we want to stay tuned for this because it probably does have practical applications in the future, but we're not quite sure right now.
There have been a couple trials, and I think they've been somewhat inconclusive, but suggesting safety, at least in populations with dementia. So I think we're going to unfortunately have to keep moving to get folks off to lunch, but it was a fantastic talk. Thank you so much. Okay, our final presentation before lunch is going to be outstanding. It's from Dr. Elena Soy from UCSF and the Memory and Aging Center, as well as the Weil Institute for Neurosciences up there. Dr. Soy is a assistant professor. She's a counseling psychologist. She got her degree at the University of Massachusetts at Boston, and she was telling us about her drive, the literal drive from Boston to San Francisco. So she's clearly adventurous. She's a bilingual neuropsychologist and a researcher up at the MAC. And much of her work has been focused on social determinants of health. And she's going to talk to us about access, accuracy, and equity in dementia diagnosis. Thank you so much.
Hello, everyone, and thank you so much, Dr. Grill. Thank you, Megan. Thank you, everybody at UCI Mind for inviting me. This is also my first time here, hopefully not the last, if I do my job right. But I'm really excited to be here and really thrilled to be joining such an amazing group of speakers. I think we've heard about so many amazing things happening with dementia research today, and also we'll hear more in the afternoon. But I'm hoping to sort of highlight some of the areas of my research, which very much focuses on access to diagnosis and especially the prevalence and the pervasiveness of diagnostic disparities that I think was brought up in one of the questions earlier today, and I would love to hear more from you. These are my disclosures. For this talk, I do not have any relevant financial conflicts of interest, and these are the research sources that I receive. So in terms of the outline, I'm hoping that today we'll get to talk about what is the current state of ADRD diagnosis, what do ADRD diagnostic disparities mean for patients, for families, for research communities and societies, what factors drive diagnostic disparities, at least the factors that we know of as of today, and what can be done to reduce disparities in diagnosis and facilitate equity. So I'll briefly start by talking about dementia as a clinical syndrome. Oftentimes many patients, I'm sure you experience this in your clinical care, they come in and they think that dementia means Alzheimer's disease, or Alzheimer's disease, means that you only have 2 more years and you're going to have memory impairment only. However, dementia is a very, very complex disease. Dementia by itself is a clinical syndrome. It is not a disease, and it has many different causes. Alzheimer's is one of them. It is the most common one, but there are many more. And dementia symptoms differ depending on the underlying cause. For example, our center sees a lot of patients with what we call atypical dementia presentations. And a lot of the patients that we see present with behavioral variant frontotemporal dementia. The first symptom in that specific type of dementia is typically behavioral changes. And oftentimes they may or may not experience memory changes over time. So this education is very important. Dementia is also very complex when it comes to clinical pathological relationships. Different types of dementia are caused by many different types of diseases. And even more importantly, many different diseases can co-occur at the same time, making therapeutics complicated, diagnosis complicated. What matters about these different types is that different types of dementia are characterized by different care and treatment needs. And as Dr. An was talking about, the behavioral and psychiatric presentations and symptoms can be very, very important across all dementias, but maybe more so in some than others. And different types of dementia are also associated with different expenditure patterns. How much a family will spend on caring for a dementia patient typically differs, as well as mortality rates. So because dementia is so complicated, dementia diagnosis is a very complex process. It typically involves many different procedures, and these are the ones that are most commonly used, but they can include more. So typically a patient would meet with an MD, with a doctor who will collect clinical history, would evaluate concerns. They might do a physical or neurological exam, neuropsychological testing that I usually do as a clinical neuropsychologist as a part of it. They can order laboratory testing to test, for example, for reversible causes of dementia. Neuroimaging can be done to look at the structural composition of the brain. And biomarker testing is also typically ordered to basically understand etiological causes, what specific disease might be causing this dementia presentation. So why timely diagnosis matters? Timely diagnosis really allows doctors, families together to work towards identification of etiological causes. As I mentioned before, because dementia is so complex and many different causes exist, identifying what specific cause, you don't want to treat somebody for Alzheimer's disease when the underlying cause of their disease is another protein. It also helps inform and coordinate care. It enables planning for the future for the families, which is incredibly important. Addressing safety issues is another important part. People start thinking about putting up bars in their bathrooms or, making sure that the stove turns off automatically. It also helps connect families and caregivers with interventions, but also education, which is key. And also helps identify candidates for clinical trials and novel treatments. So when we talk about timely diagnosis, at least as of now, we're really talking about that stage of dementia that we typically might refer to as something like mild cognitive impairment or mild behavioral impairment in the case of behavioral variant FTD. So what is the current state of dementia diagnosis across different communities? First of all, dementia is massively underdiagnosed, especially when it comes to its earliest stages. Recent reports indicate that patients who have not received a dementia diagnosis account for 50 to 66% of all cases in primary care samples. And I think this is incredibly important because we don't want to say that, primary care physicians do not recognize dementia. I think it's more so that we have to understand these disparities in the context of existing systems that I will talk about later today. And underdiagnosis has been studied in primary care samples as well as in neurology clinics, and different types of research has been done, qualitative, mixed methods, quantitative, but most research agrees that underdiagnosis is more common among patients who identify as non-Hispanic Black or Hispanic ethnicity, have lower educational attainment of less than high school, have below median wealth, have limited English proficiency, and do not have a partner, a caregiver, or informant present with them. So dementia, even when recognized, is typically recognized very late in certain communities. This is our own study published just recently, where we looked at over 11,000 Medicare beneficiaries across California. And we tried to understand what are some of the factors that underlie late diagnosis. So the first point of diagnosis occurs at the time when the patient has already progressed enough to meet criteria for full dementia and not mild cognitive impairment. And we found that within this Medicare sample, late diagnosis was more common in beneficiaries who identified as Asian, non-Hispanic, Black, and Hispanic. They were older at the time of their first diagnosis, and they also had a greater medical comorbidity burden. Even more importantly, we found that when people get a diagnosis of dementia, their diagnostic evaluation was less comprehensive. So they did not receive the elements that are typically involved in that list that I showed you before, like identifying reversible causes, seeing a specialist, or getting neuroimaging done. And this was more common in Asian and Hispanic older adults. And these are just some staggering numbers, descriptives from our study showing that 23% of non-Hispanic white beneficiaries were diagnosed at the stage of MCI at a timely diagnosis, compared to 18% of Hispanic beneficiaries, 16% of Black beneficiaries, and only 12% of Asian beneficiaries. Another study that I'm running right now that I'm hoping to publish this year looks at identification of a specific diagnosis. You know, kind of going back to this idea that dementia can be caused by many different diseases, it's important for us to be able to identify what is the cause. And that etiologically specific diagnosis drives care, you know, consultation with the family, expenditures, et cetera. And so in this sample, where I'm looking at almost 900,000 Medicare beneficiaries across the country, who have a diagnosis of dementia, we find that non-Hispanic white beneficiaries in general constitute about 80% of all cause dementia. But as we are looking through more and more complex or less recognized types of dementia, that number increases. For example, among all patients with frontotemporal dementia, non-Hispanic whites represent 92%. You know, imagine just the current constitution, demographic constitution of older adults, and how significantly over or underdiagnosed, however you want to think about it, frontotemporal dementia is across these groups. So diagnostic disparity has profound effects on patients and families. Diagnosis is typically the first step in getting all the additional elements of dementia workup, of dementia care, of dementia consultations. And so you can see in this fantastic study published just recently in Alzheimer's and Dementia that diagnostic disparities occur at the time of initial assessment, but because they occur, we then see that there are effects on ongoing treatment and support. For example, people who are underdiagnosed or misdiagnosed or diagnosed late are less likely to receive and more likely to discontinue anti-dementia medications like donepezil and memantine, and likely other medications that we heard about from Dr. Pierce today. There's increased antipsychotic use. There's increased hospitalization and emergency room utilization. And also sort of later on, we also see profound disparities in terms of accessing long-term care and hospice care. And I think it's even more relevant when we talk about these new drugs, right? It's a very exciting field. It's sort of developing these new drugs and developing these new potential cocktails of how we're going to treat different types of dementias maybe altogether. We likely agree that in order to be able to eligible and benefit the most from these drugs, people need to be diagnosed timely. Early, we need to be able to identify etiological causes so accurately. And, you know, with this approval of these medications, you know, people were very excited when CMS said, yes, we're going to cover these medications, but look at the actual diagnostic. To be able to be eligible for the coverage, you need to be diagnosed with mild cognitive impairment or mild Alzheimer's dementia with documented evidence using biomarker testing, right? And then another important thing that I always like to highlight, you know, as a clinician, as I'm sure you all are, I think it's very important to hear from families and patients about what timely diagnosis means to them. And in a fantastic qualitative study also published very recently, there are caregiver reports about what early diagnosis or timely diagnosis really meant for them. And this is a spouse of a who identifies as Hispanic and lives with his wife, who also identifies with Hispanic, who said, well, it was, I would say, good, not good, but it was something that at least we finally found out what it was because we were not sure what the problem was. Now we know how to deal with it. Another caregiver said, have I not had the diagnosis, that would be a disaster. So at least I know, based on that diagnosis, what road I was going down. Again, it's this ability to empower families and, giving them agency and participation in this process together. So diagnostic disparities also have a profound effect on research community. You can ask every single researcher in this room and we will say, yes, this is a significant problem. Why is it a problem? It's because late and misdiagnosis typically are associated or likely underlie significant underrepresentation and sometimes exclusion of diverse older adults in clinical trials, epidemiological and observational trials, because we really cannot include somebody if we don't have an appropriate referral. This pervasive pattern may in turn lead to biased results in the studies that we run, in the conclusions that we derive, and then these results and conclusions influence future studies because you need the evidence base to run anything new. And just as an example, in a paper published in Brain a couple of years ago, they looked at the sort of overall constitution of all of the participants who were included in the Alzheimer's disease GWAS studies, genetic studies, as we heard about before. And non-Hispanic white or European individuals who have European ancestry, gosh, I always get confused with my methodology, but individuals with European ancestry in general constitute 16% of the world's population, but they represented over 60% of all individuals included in the Alzheimer's disease GWAS studies. So what do we know about the genetics of Alzheimer's disease is limited to a very limited group of patients or future populations. So I think that is an incredibly important gap to address. And finally, diagnostic disparities also have profound effects on economy and society. Because we know that older adults who experience diagnostic disparities are often exactly the communities that are at greatest risk for dementia. I want to give you an example. For example, these are all of the factors that have been identified as being associated with diagnostic disparities: being non-Hispanic Black, being Hispanic, having high comorbidity, or being socioeconomically disadvantaged. Look at the actual prevalence and risks for dementia. Prevalence is twice as high in Black Americans and 1.5 as high in Hispanic Americans. Incidence is highest in Black and Hispanic Americans. And age, low education, and high cardiovascular risk burden each increase dementia risk. So if we are not treating patients who are at greatest burden and will also likely benefit the most from these medications, what are we really doing with our studies and how are we able to continue developing effective methods? So in this study that was done by Alzheimer's Association, it was a simulation study that found that if 88% of individuals are diagnosed in the MCI phase, $7 trillion could be saved in healthcare costs. Most of these were associated with Medicare savings, and these were due to smaller spikes in costs immediately before and after diagnosis, and also lower medical and long-term costs. But I, again, want to emphasize that these potential benefits, social, economic, family, would not be realized unless diagnostic disparities are addressed in a timely manner. So what drives diagnostic disparities? Really, the mechanisms underlying many different types, not just Alzheimer's diagnostic disparities, healthcare disparities in general, are very complex. They're likely multifactorial and are not very well understood. However, to date, research has identified at least two broadly defined areas of where diagnostic disparities may arise: patient-level factors and systems-level factors. I'll first talk about patient-level factors, and these will likely be things that would resonate with a lot of clinicians. For example, language and communication barriers present a significant challenge. 48% of US adults with limited English proficiency encounter language barriers within their healthcare in the last 3 years. And if you look at the actual areas in which these difficulties occur, like filling out forms for a doctor or communicating with a doctor or understanding instructions about how to take medications or what the diagnosis is, I think are incredibly important, especially when we talk about a condition as complex as dementia, that can have many different causes, is associated with many different types of testing. This is likely one of the factors. Another important aspect of it is health and healthcare literacy factors. You know, there is some research indicating that there is lower awareness of dementia in certain communities. Again, this is all published by Alzheimer Association, and there are surveys, population level surveys of the country. They found that more than half of Asian, Black, Hispanic, and Native American participants believe that significant loss of memory or cognitive decline is a part of normal aging. There is also a limited trust in healthcare systems and services, including medical research, among certain communities. Substantial number of Black individuals, Hispanic individuals, Native American, and Asian individuals believe that it is harder for them to get excellent care for dementia because of their race and ethnicity. And I think this is incredibly important, and it kind of made me think about Dr. Turner's talk and how, you know, this idea about health literacy kind of goes beyond just sort of understanding a certain disease. It's more understanding disease within the systems within the disease occurs. And then another important aspect of sort of understanding cultural influences on dementia diagnosis are the perceptions of dementia and aging and potential stigma of mental illness in certain communities. These are very, very complex phenomena. They are not well studied. I think we need large qualitative / quantitative studies to better understand how the stigma arises and how it's potentially perpetuated. But I think there are some qualitative studies that really highlight, you know, what is it that matters? And within certain communities, you know, that fear could be driven by loss of dignity or changing the perception of who you are as an older adult within your family systems. The fear of being a burden to your family if you get diagnosed, you know, what does that mean? Maybe, you know, a fear of being unable to contribute to society over time because this diagnosis is so definitive. So these are all very, very important factors to consider, and also for clinicians to be aware of in order to be able to provide culturally informed care. I also want to talk about race, right? In my previous slides, we saw that, you know, race was associated with many different types of disparities, but I do want to emphasize that race is a social construct. It has absolutely no biological basis, and genetic factors do not account for the large differences that we see in dementia diagnostic disparities. As a matter of fact, I think most my epidemiology friends would not let me lie, but within social epidemiologic sort of framework, you know, race is typically associated with a lot of different socioeconomic disparities that occur early in life, in middle life, and in late life that they then can drive differential access to healthcare and differential access to diagnosis. I also want to highlight that using race in research and also in terms of understanding disparities is incredibly important, but it's also important to understand within race variability. For example, Pew Research Center finds that Asian Americans are the most socioeconomically diverse group in the country. So if we just put everybody under the same category, we are missing so much more or masking disparities that might exist within specific communities. Same thing with Hispanic Americans. You know, you ask anybody, like, you know, what is a Hispanic American, right? There's so many different ethnicities, cultures, languages, socioeconomic conditions. And I think it's incredibly important for us to be attuned to that, to get to that precision medicine standard that we're all trying to get to. I want to emphasize more that again, take it with a grain of salt, but I think system level factors are much more important and also are much more actionable when we think about solving diagnostic disparities. So system level factors typically refer to historical and structural systems that have been established within healthcare and outside in the broader society that really guide the way that the patient might navigate their dementia journey. So one of the most commonly sort of encountered challenges is the lack of culture appropriate services and tools. As a neuropsychologist, I can say that there are probably a handful, like I can count them on one hand, of tests, neuropsychological tests that are definitive for dementia diagnosis that have been validated in populations other than non-Hispanic white English speakers, right? It's even more challenging when you think about working with primary care physicians who have an even more limited time in order to be able to identify these tools, and administering them and scoring them and interpreting them is another challenge. There's also a profound shortage of dementia specialists, especially in low resource areas, both within this country and internationally. This is another recent study that was just published looking at the dementia diagnosis intensity around the country. And it showed that where you live might actually increase your chance of getting a dementia diagnosis twofold. And I think it's incredibly important because you can look at these red dots that you probably can't see, but the red dots indicate Alzheimer's disease research centers, where they are in the country. And I think you can sort of see a little bit less of that variability within when there's a close proximity to a specialty service. Another important factor to consider is there is inadequate training of general healthcare professionals, GPs or PCPs, in the recognition of dementia. There have been several studies done, but I really like this one that looked at, they surveyed about 300 PCPs around the country, and they tried to understand what are some of the areas in which PCPs feel less confident. And 21% of PCPs said they were highly confident that they correctly recognized when a patient had an NCD, so only 20%, and only 13% were highly confident in making a specific diagnosis. There's also potential bias in referral practices. In a huge healthcare system down south, there was a research looking at their medical records that found that African Americans were less likely to be seen in specialty memory clinics and received most of their dementia care within primary care. But that also meant that they didn't really have access to those high-level services, like, for example, PET neuroimaging. Okay. And there are also time and cost related barriers, of course, talking about insurance coverage, transportation costs, everything that we experience in our daily life. So is this all doom and gloom? No, I think there's a lot that we can do, and there are many different approaches that we can think of in terms of improving access to dementia diagnosis. And I think there is no definitive answer, but a multi-pronged approach would likely work the best. So on the level of communities, it is important to have or to establish and sustain culturally tailored campaigns to increase public knowledge about brain health and dementia. And Dr. Schindler will talk about new biomarkers today. We get asked about them all the time. And I think having that community level accessible education is incredibly important. It's also important to have culture and location informed campaigns to facilitate the navigation of dementia care. Just look at this diagram that shows a typical journey through, from dementia diagnosis to actual care is incredibly complex. And I think the more we can do to support the communities that we work with, the better. Within healthcare systems, increasing the diversity of general practitioner and dementia specialty workforce is incredibly important. It's important to have, I think, a level of concordance between whether it's cultural, linguistic, or maybe race, ethnicity related between the doctor and the family, because having that cultural knowledge really enhances the family's trust and also their ability to understand the disease better. Better education and training for general practitioners on dementia recognition and culturally informed care is definitely needed, and I'm hoping this conference is one of these examples. And also, I think, outreach by dementia specialist providers to facilitate the referral of underserved groups. I have been working with primary care providers in Southern California and in the Bay Area for the past 6 years. And this has been the most incredible experience for me to learn about where the gaps are in primary care and what we can do as specialists to help connect us together. And finally, policymaking. It's incredibly important it's very difficult to make any of these changes without appropriate policy support. So providing adequate reimbursement and time for providers to diagnose and manage dementia is important, and also improving access to effective dementia care models that provide critical support, not just to individuals, but their carers and general practitioners following diagnosis. An example of this model, I think, is GUIDE that Dr. Gibbs is going to talk about later today that we're all very excited about. But I want to stress that without having access to effective dementia care models, diagnosis by itself will not be sustainable. We have to make sure that diagnosis comes with the actual plan of what we can do for the patient. So here are my conclusions. There are substantial and persistent disparities in availability, timeliness, and accuracy of dementia diagnosis. These inequities have detrimental impacts on patients, families, and societies that we live in, and collaborative efforts among researchers, clinicians, community partners and policymakers are needed in order for us to make a dent in these disparities. And also, Dr. Gill, can I just quickly, for a second, I just wanted to quickly plug a couple of training programs that we have at the UCSF Memory and Aging Center. We're really excited to run more of the equity programs. My lab is entirely focused on equity. And if you're interested in a postdoc, a 2-year postdoc, T32 funded, in equity and brain health, we have a lot of opportunities to work on studies similar to mine or any other study that you're thinking about. And there's also a fantastic training opportunity for international scholars that we have called Atlantic Fellowship in Brain Health. This is typically for individuals in their mid-career coming from all over the world, and we really want to help them navigate their career and establish dementia care in their communities. So please feel free to reference these QR codes. Thank you very much.
Time for a question or two. Got one over there. We need a microphone though.
Diagnosis, like we have MOCA or MMSE, take a lengthy amount of time that may not be available at clinic appointments. Are there free or low cost, like maybe government resources that we can, as a patient, to be aware of to go to facilitate diagnosis and management?
I'm sorry, you mentioned MOCA and MMSE,
No,
and
I was saying that diagnosis takes a long time, and so it might be hard for patients to kind of schedule with the cost of doctor visits, can be high for patients, sometimes inhibited. Are there government or other free or low cost resources that patients can go to kind of get the diagnosis and the dementia management to make it easier and more within reach for more people?
yeah, so I think it's a great question, and I think it really depends, right? So when we think about, again, even access to primary care can be a challenge. I think what you're highlighting is that, even primary care providers are not necessarily a resource that is available to everyone. So I think it's incredibly important, those community-based interventions that I talked about, I think they help facilitate, again, not just sort of understanding that, dementia is important, but also how do I request those services, right? Like, how do I connect interventions? How do I find a primary care physician? What do I need to talk to with my insurance? I think those are very important pieces, but I'm not aware of any sort of government level programs yet. I would say yet. I'm hopeful that they're coming in the future.
I see one more question, but maybe we can connect you with that person as we get folks to their lunch. That was fantastic. Thank you so much. So enjoy your lunch. We'll be back here right at 1 o'clock. Okay, here we go. It's a time of fantastic progress, enthusiasm, excitement, advances in what we're learning and how it affects clinical practice. And I am sure you have seen headlines about blood tests as one particular area of incredible excitement and enthusiasm about that progress. And we have one of the world's absolute leaders on the development of blood tests for Alzheimer's disease, Professor Suzanne Schindler from Washington University and the Knight Alzheimer's Disease Research Center. Dr. Schindler has done some of the seminal work about these blood tests and their applicability to making the diagnosis of Alzheimer's disease, and relevant to earlier talks, whether those things differ among particular communities. She came to WashU in 2002, and as she told me last night, she never left. So she's done a lot of her training at the Eminent Center there at WashU, including her fellowship with Dr. David Holtzman, whose lab, really was critical to the development of the drugs we heard about earlier today too. She's received numerous grants and prestigious awards, and she's really a leader in our field. So we're grateful to her too for joining us today, and she's going to talk about blood tests for Alzheimer's disease.
Well, thank you so much for inviting me. And this is my first conference, but I could see why it's gone on for 35 years. And, very impressive what you have done here. And I'm sorry to tear you away from lunch. It was very tasty, but I'm gonna get going. at the point in my life where I have to figure out whether I need my glasses or not. So I am a clinical neurologist and dementia specialist. And so I'm going to approach things today from that kind of perspective. And I'll tell you some about blood tests. I'm not going to get into the technical weeds that much, but I'm going to tell you about how these are relevant to you and where the field is going. These are my disclosures. Notably, I have not received any personal compensation or direct research funding from any diagnostics companies. So I like to start my talks about diagnosis with the most common question that I'm asked by my patients, which is, what is the difference between dementia and Alzheimer's disease? And we heard some about this earlier from Dr. Soy and others, but I think it's really important to clarify at the outset that dementia is really an umbrella term that refers to a decline in memory and thinking that impairs function in everyday activities, and that there are many, many causes of dementia, not just Alzheimer's, but all sorts of different issues can lead to memory impairment. And Alzheimer's is a specific brain disease, and it's defined not by the symptoms, but by the brain pathology, these amyloid plaques and tau neurofibrillary tangles. Now, Alzheimer's disease pathology is the most common cause of dementia in individuals who are older than 65. That's why they get conflated sometimes, but they're really different concepts, where dementia is the symptoms, Alzheimer's disease is the pathology. And it's also important to remember that many patients with Alzheimer's disease pathology have all sorts of other conditions that can additionally impact their cognition. And that's really important to understand as a diagnostician, that even if someone has Alzheimer's disease pathology, they can have all sorts of other things going on as well. So when patients come in to see me, a major question that is often asked is there cognitive impairment due to this Alzheimer's disease pathology or something else? And the traditional evaluation that we have done for dementia in many ways hasn't changed for decades. And this has really been oriented to essentially ruling out non-Alzheimer's disease causes of cognitive impairment. And so we do a detailed clinical history, trying to talk to someone who knows the patient well and has known them well for years to see if there's been a significant decline. We ask about things like their medical history, medication history, family history. We do objective cognitive testing and a neurological examination, routine blood work, and structural brain imaging. Again, a lot of the reason why we do these things is to try to rule out other things that could be causing their symptoms. But interestingly, for many, many years, we've had good tests that tell us whether someone has Alzheimer's disease pathology, and we have not used them. And I'll kind of tell you why. But for many years, Alzheimer's disease has really been, in many ways, a rule-out diagnosis, and that is changing. And one reason that's changing is there's really been increased awareness that if we want to diagnose people accurately, we have to use these things called biomarkers. And I'm going to go into detail about what biomarkers are, but really what they do is they tell you whether this Alzheimer's disease pathology is present in the brain. And the reason why these are important is that we have learned that even when we go through that full evaluation that I showed you, which is comprehensive, takes a long time. This is part of why dementia specialists are hard to get in to see, and why diagnosis is delayed. Even after we complete that whole evaluation, the diagnosed cause of cognitive impairment is often wrong. And being a dementia specialist is rather humbling, because when we do these biomarker tests, sometimes we find out, not infrequently, that even patients that we think have a very typical AD dementia syndrome, they don't have Alzheimer's disease. they have cognitive impairment due to something else, but it's not due to this Alzheimer's disease brain pathology. And so we have really learned that these biomarkers are essential to telling us whether this pathology is present in the brain and may be causing or contributing to cognitive impairment. So what are the indications for clinical AD biomarker testing? And I think this is important to go through for a couple of reasons. We do not just do biomarker testing willy-nilly on people. We only typically do biomarker testing on individuals who are seen, have already had this comprehensive workup that I described, and have objective evidence of cognitive impairment. And it is currently not recommended to do biomarker testing on individuals who are cognitively unimpaired. I'm not going to get into all the reasons why, but it's somewhat related to what we heard about for why we don't do ApoE testing on individuals. To do biomarker testing for Alzheimer's disease, we have to think that Alzheimer's disease is a potential cause of cognitive impairment. And we also have to think this biomarker testing is going to help the patient in some way, that it's going to affect our diagnosis, or that it may affect our management. So for example, if I have a patient where I don't think the test is going to change anything at all, then I'm not going to order it. So we heard from Dr. Pierce a little bit about these biomarkers. So there are actually lots of different ways to test people to see whether they have these amyloid plaques and neurofibrillary tangles in the brain. But the three modalities that we typically use in our clinical practice are amyloid PET, CSF biomarkers, and blood-based biomarkers. So I'm going to talk a little bit about each of them, but then I'm going to go into more detail about these blood-based biomarkers. So to perform an amyloid PET scan, you inject a patient with a radio tracer that goes up into the brain and actually binds to the amyloid plaques. And then you do a PET scan, and essentially the plaques light up, and you can take a picture of that. So this is a great technique. It tells us a lot about how much plaque is in the brain, where it's located. But the issue about this clinically is that it's expensive to do PET scans, and these radioligands are expensive too. They're usually around $3,000 a dose. And you have to have really specialized equipment, personnel to do all of this. And so the cost is around $6,000 a scan, more if you're in Southern California, I've heard. This can sometimes lead to significant co-pays for patients, and some patients are concerned about radioactivity. So these are some significant drawbacks. And unfortunately for PET scans, I think these are drawbacks that we can't fix. So there's not going to be a way that we can make amyloid PET scans cheap and easy. So the next modality is CSF biomarkers. And this has been the way that we've done most of our biomarker testing for years in patients. This requires doing a lumbar puncture or spinal tap to collect cerebral spinal fluid. Patients actually tolerate this pretty well. And we can measure the concentrations of certain proteins in the cerebral spinal fluid, and there's specific patterns in the changes of these proteins that tell us whether someone has these plaques and tangles in their brain. And the results of the CSF biomarkers and the amyloid PET scans agree quite well, about 90% of the time. And when they don't agree, it's usually people who have borderline levels of pathology. However, there are drawbacks. So most clinicians do not perform lumbar punctures, and it's time consuming. It's expensive to train staff to set up these clinics and perform LPs. And patients are not typically excited about getting a spinal tap. Some refuse because they're worried about risks or perceived risks. And unfortunately, we see that individuals who identify with minoritized groups are much less likely to undergo LP. So the use of CSF biomarkers really creates disparities in diagnosis, which we heard about from Dr. Soy. So now let's talk about blood tests. So with blood tests, you collect blood, just like you collect blood for all sorts of things in general medical practice. And we perform analyses very similar to what we do with CSF tests. They just have to be a lot more sensitive. And we have found that some of these blood tests, as I'll show you, are actually as accurate as FDA-approved CSF tests. However, there are some drawbacks. This includes that right now, insurers typically will not pay for these tests, or sometimes they pay and sometimes they don't. Some of these tests, as I mentioned, are very accurate, and some of them are very inaccurate, not much better than a flip of a coin. And we have learned through work by Michelle Milkey and others that some of these tests are affected by medical conditions like chronic kidney disease that can lead to false positives. And also, as we've heard from Dr. Soy and others, we have not really validated a lot of our research findings in diverse groups. And some of the work that we have done has indicated that some of these tests may not perform consistently in all racial and ethnic groups. And this is a big problem because, of course, we don't want to disproportionately misdiagnose one group. That would be very unfortunate. So I talked about 3 different modalities of biomarker testing. And right now, where I work at Washington University, we're using all 3 modalities. So this plot is showing the number of tests on the y-axis and time on the x-axis. And this is our biomarker testing over the last few years. Initially, we were just doing CSF tests. That's because what was available, that was what was reimbursed by insurance. We were doing about 6 tests per month. Then blood tests became available, the first generation Passivity AD tests. And partly because it was developed at WashU, we felt comfortable using this because we knew how sensitive and specific it was and when it was appropriate and when it wasn't. And so we started using that in some patients, typically those patients who couldn't get spinal taps for whatever reason. And that allowed us to increase our volume of biomarker testing. And then really quite recently, Medicare approved payment for amyloid PET. So we've had amyloid PET for a long time now, not quite 20 years, but quite a long time. And it's been FDA approved for at least a decade, but we have not used it because Medicare wouldn't pay for it. But now that we have these new therapies, Medicare started paying for it, so we have started using it. So you can see that we've had a big uptake in our rate of amyloid PET scans. We also, you may notice that our blood tests went up, especially before the amyloid PET scans. That's because we got a more accurate blood test, the 2nd generation Passivity AD2 test that includes this really important protein that I will mention called p-tau217. And so we felt more comfortable using it. So it's been very interesting watching in real time how all of this biomarker testing is really affected by these really practical sort of factors. So we've increased our biomarker testing about 7-fold over the last 3 years. I do want to emphasize the Alzheimer's disease blood test that we're showing here, this is not research, this is clinical care. We're using this in clinical diagnosis and decision-making, being aware of the limitations of these tests. And we found that the modalities that we're using are greatly affected by practical considerations such as reimbursement, also the accuracy of the tests. So it's really been interesting watching all these changes. I would point out that lecanemab was approved in July of 2023. We started increasing our testing before lecanemab was approved. We're getting patients teed up. And then we've increased our testing even more. So a lot of the reasons for these shifts in patterns are these new treatments. So this is looking at how biomarker testing has been performed in the past and right now at this moment, but what's going to happen in the future? So my very strong opinion is that clinicians and patients are going to need Alzheimer's disease blood tests in this new era of Alzheimer's disease treatments. So we've been hearing about some of these anti-amyloid treatments this morning from Dr. Pierce, but there is a very loaded pipeline of treatments that are specific for Alzheimer's disease. So there's nearly 150 different treatments that are in trials right now. And I'm going to bet that some of those are going to work. And many of these treatments will require that we confirm someone has Alzheimer's disease before we start them. So it's not just going to be anti-amyloid treatments, it's going to be other treatments that require this as well. And so over the last 5 years-ish, there's really no good numbers on this, but I would guess that maybe there's been 20,000 AD biomarker tests performed yearly in the US. That's just a rough guess. But I think with this new era of treatment, we're going to need to do hundreds of thousands to millions of biomarker tests to make sure that everybody who needs a test can get a test, and really to support access to new treatments. And I don't see that that's going to happen with CSF or amyloid PET. There's just no way to scale up those modalities. It would be very, very challenging to do that. But we could do that with blood tests because blood tests are less invasive, they're more accessible, they're less expensive. And also I would just point out that these blood tests are, because they're more acceptable and accessible, we think that their use may help to reduce some of the disparities in Alzheimer's disease diagnosis and treatments that we've heard about. So there's really a lot of positives to this. So also positive is I think we have the tests that we need now. They exist now. So this on the left, this is showing a test that was developed at WashU by Nico Bartolomei in the laboratory of Dr. Randall Bateman. And we've found that this protein, p-tau217, or more precisely, this ratio of phosphorylated to non-phosphorylated p-tau217 is highly accurate in classifying whether someone has amyloid pathology in their brain or not. And when we do a head-to-head study, it's just as accurate as FDA-approved CSF tests. And this is the test that was licensed to C2N Diagnostics and is now Procivity AD2, and was recently demonstrated to perform very well in a Swedish cohort. And one thing I would note is that a concern about some of these blood tests has been that they may be affected by medical comorbidities. And what they found in this Swedish cohort is even in cohorts that had quite a high rate of medical comorbidities that you can see there on the bottom right. So we're talking chronic kidney disease of 26% in primary care, 17% in primary or in secondary care. Cardiovascular disease rates of 70% in primary care, 48% in secondary care. These tests are still performing very, very well. So that's overall reassuring. At the NIGHT ADRC, we have been very concerned about the performance of these biomarker tests across different racial and ethnic groups. And one of my colleagues, Dr. Kenji Zhang, actually has a large grant that has brought together multiple ADRCs to see what biomarkers do in Black individuals and white individuals. Do you get the same results? Do you get the same correlations between cognition, for example, and these fluid biomarkers? Now, we're still running a lot of these samples, but at least in our preliminary analyses, we find the results very promising, that it looks like we're getting high accuracy and very consistent results in both Black and white individuals for this particular test. And that is what's interesting about this test is it's a combination of plasma A-beta 42 to 40 and percent p-tau217. So it's a combination of two ratios. This is different than some other tests that just look strictly at concentrations. And again, I won't get into the technical weeds, but we think that some of these ratios really may help to mitigate some of the associations between biomarker levels and things like chronic kidney disease. And this has actually been shown in some work. However, that's just one test. There's a lot of other tests out there. And so I think someone was asking earlier, like, how do I get this test? Well, it turns out that there are at least 5 tests that your doctor can order today. It seems like more and more tests are coming on into the market all the time. The problem is that some of these tests are accurate and some of them are not. So I am part of a group, the Foundation for the National Institutes of Health Biomarkers Consortium, working with a large study called the Alzheimer's Disease Neuroimaging Initiative. We did a head-to-head study of 6 of the leading blood tests or blood test panels for Alzheimer's disease. And so on the upper left, you can see the company platform name. What's significant is 4 of these are tests that your doctor can order today. The top 2 by C2N and Fuji Rebio are available from Mayo Clinic Laboratories as a send out. So these exist today. And what we find is that some of the measures perform very well. So just to briefly describe this, the closer the number is to 1, the better. And really, all the tests that are dark green here are really performing very well, very similar to what we see with CSF tests in classifying amyloid PET status. Those dark green bars are p-tau 217. And so we think that really all 4 of these p-tau 217 tests will do quite well in helping us to diagnose patients with cognitive impairment, whether they are likely to have Alzheimer's disease brain pathology or not. However, some of these other tests did not do as well. So look in particular at the blue, which is A-beta 42 to 40. Some companies are offering just A-beta 42 to 40, and that measure is not nearly as good at classifying amyloid status. So an issue here is that some providers don't know which test to use, don't know which measures to use, because it's A-beta 42 to 40 and there's an amyloid in the name. They think that's the best measure of amyloid when really it's p-tau 217 that's the best measure of amyloid. So there's a lot of education that needs to happen. Also interesting is that we find that these p-tau 217 measures don't just best correspond to amyloid status, they also are the best predictors of tau PET. They are also the best predictors of cortical thickness and cognitive impairment. So across the board, p-tau 217 is really doing very well. So I talked about, we have a lot of tests out there. There's a lot of confusion about how to use these things. How do we tame this wild west of 80 blood tests? So we've actually been thinking about all the steps that need to be done here. So one is setting minimum acceptable performance standards for the tests that we use in clinical care. And so we had a pretty large group that was coordinated by the Global CEO Initiative that brought together academics, industry, nonprofits to talk about what's acceptable in terms of these tests, what's not acceptable. And what we decided is that these blood tests need to have 90% sensitivity and specificity for amyloid PET status. The reason we chose that number is that's what FDA-approved CSF tests have. So it didn't make sense to force the blood tests to have a higher threshold of accuracy than the CSF tests. So that's the threshold we set. Again, that discordance that we see is most often in people who have borderline levels of pathology. And there's other thoughts about how to deal with that group. You can use blood tests that are not quite as accurate if after you do that blood test, you do a second test, like a CSF test or an amyloid PET scan. And we published a paper with these consensus recommendations. Have to see whether people follow the recommendations or not, but we have at least set the standards. Right now, we're trying to define which specific tests meet these standards and their appropriate use. So the Alzheimer's Association has an appropriate use recommendation committee and is carefully reviewing all the literature on all of these tests, and will make recommendations about which tests are meeting those thresholds and which tests aren't. And also the FDA has received applications from some of these diagnostic companies for some of these blood tests. And they're reviewing this. We'll have to see. It's a government agency, but I'm hopeful that in the next year or two that we will have FDA-approved blood tests for Alzheimer's disease. And when that happens, I think that payers will essentially be forced to start paying for these things. And maybe because amyloid PET scans cost so much, they'll be happy to pay for these things. We'll see. There is another big issue though, and that is that in the past, the only 80 biomarker tests we had were amyloid PET and CSF. And that made them very onerous to use. And so only dementia specialists used them, which in some ways was good because dementia specialists had a lot of training on what these tests meant. Now, these Alzheimer's disease blood tests are pretty simple, and it means that primary care docs can order them, which in a lot of ways is good because I don't know if you've heard, but sometimes it takes 3 months, 4 months, 5 months, 6 months, a year to get in to see a dementia specialist. And we really want to arrive at a Alzheimer's disease diagnosis much earlier than we did in the past. But there is some danger here in that it's really important that people understand not just how the test performs, but that you have to integrate it with clinical information to arrive at an accurate diagnosis. And I'm, again, not going to get into the weeds here, but basically, if you have a positive result in a patient who has a high pre-test probability of having amyloid pathology, you can trust it. If you get a negative result in a patient who is less likely to have amyloid pathology, you can trust it. But if you get discordant results that don't make sense, you need to think about this again. What this means, though, is that you have to formulate a clinical impression and think about whether this test result makes sense. You can't just use it as a standalone. So looking forward, I really expect that these blood tests within a relatively short period of time are going to be pretty broadly used in the clinical diagnosis of cognitively impaired patients. And what happens after that? Well, we've heard about a couple of these prevention trials like AHEAD 345 and Trailblazer 3. We'll have to see what these trials show, but if they're positive, it means that we may need to start screening cognitively unimpaired older adults with these blood tests to see whether they might be candidates for preventative treatments. Now, that's still a little bit away, but it does give me hope that we could enter an era when high amyloid and tau in a cognitively unimpaired person could be managed preventatively, kind of like what we do with high blood pressure, high cholesterol. We all are looking forward to the day that we end Alzheimer's, and this gives us a possible way to get there. So I would like to thank our clinic patients, research participants, and their supportive families, my colleagues at Washington University, especially Joy Snyder, who's the director of our memory clinic, and my longtime mentor, Dr. John Morris. Thank you.
Excellent. Time for a question or two.
Hello. Let's see if I can make sense of my thoughts. I think I really appreciate the value that biomarkers can bring to the diagnostic accuracy and also treatment and management for dementia. But I always get a little worried when I hear the excitement about blood biomarkers as potentially talked about as alternatives and easy ways of diagnosing Alzheimer's disease. And it just frightens me a little bit because I think about misuse where that gets replaced and in the light of saying, okay, let's work on diagnostic disparities and just administer blood tests across the board and use that in a way that it screens out people for further evaluation with more comprehensive ways of evaluating. So that's what scares me. And if you can speak to that a little more. I know you did touch on it, but.
Yeah, no, I think that's a very important and valid issue. I think there is a danger that when you have a test that people just do the test and they don't think about the clinical scenario, they don't integrate it with clinical data. So we're really going to have to push on clinicians that they have to keep being a clinician and treat the patient and not the test. So yeah, you're right. I think that there is potential for misuse there. Again, we're just going to have to keep educating people to how to deal with that and keep reinforcing that dementia is not just caused by Alzheimer's disease, that there's many causes of dementia, and that even people who have positive biomarker tests, they often have other conditions that are contributing to their cognitive impairment, and you have to keep looking for those. And my patients with Alzheimer's disease, I can make them better if I find out that they have sleep apnea or if I stop their medications. So we have to keep being doctors. We can't just test people and be done. So thank you.
The statistics show that you have a tremendous amount of patients who develop the Alzheimer's disease actually, and the tests are just being tested in 2024. The cost of Alzheimer's treatment is $60,000 a year. And the study so far right now has come to 18 months follow-up. So I'm going to be able to see if I get tremendous disparity between the numbers of patients who do actively have Alzheimer's disease and numbers of Alzheimer's patients who get treated. How can you solve the disparity of treatment and patients?
So I'm sorry, I was having a little bit of.
I think the question is that the new treatments are very expensive, and that there is a great risk of disparities by who will be treated with these new treatments and who will not.
Okay, thank you. So I think that's absolutely correct. And we see that in our own clinic, that the people that are getting on these treatments are very well educated and well-off individuals. So I think that's absolutely true. I guess I have a couple thoughts about that. One is that the lecanemab and infusions generally are a lot of work, and we're developing the infrastructure as we go. And so in some ways, it's good that people have more resources are helping us to trailblaze here. But we're going to have to get to a point where these are available to more people. And something that actually gives me hope is that pipeline that I was showing you, because there's a lot of drugs in the pipeline that are not infusions. And I think that the work that we're doing right now, setting up treatment clinics, is going to help us with those treatments as well. But yes, you're right. There's big disparities right now, and it's going to be hard to fix that.
Unfortunately, I think we need to keep moving. So we're going to have one more talk before a break, and maybe the folks who have questions could find you at the break. Thanks for a wonderful talk. So the other good news about the payment for those medications is that Medicare is reimbursing for those drugs. And the last talk before the break is about something else Medicare has begun reimbursing for, which is Dementia Care Navigator programs. And we're very excited that our own Dr. Lisa Gibbs, who is a professor in the Department of Family Medicine in the UCI School of Medicine. She's the Ronald Reagan Endowed Chair of Geriatric Medicine, Chief of Geriatric Medicine and Gerontology at UCI. I know she's taken on other administrative responsibilities at UCI that are beyond my list here. But importantly, she was the Physician of the Year for the Orange County Medical Association in 2017. She's a fantastic colleague, and she is helping lead up UCI's efforts to be a part of, there you are, Medicare's coverage for Dementia Care Navigator programs through the GUIDE program. Thank you so much, Dr. Gibbs.
All right, thank you. It's a pleasure to be here again. Thanks to Mind for sponsoring this event so that the dementia community can come together every year. All right, so we're all teed up. So I want to talk today, we're going to switch gears a little bit and talk about the care and support of people living with dementia. It's taken a lot of hard work over a lot of decades from healthcare as well as advocacy organizations to really keep dementia and other related dementias on the attention and the minds of the people that make major decisions in this country. The good news is, as Josh mentioned, we're finally getting acknowledgement from one of the largest healthcare insurers, Medicare, that we cannot care for patients with dementia without some major reform. And I will talk about some of those and then move on to the guide program a little bit later. All right. Okay. I'm going to talk about one reason why I think that this progress has been so slow, and that is ageism. I'm going to move on and talk about some of the current and future threats to care delivery, which is our workforce. And then I'll talk about some of the new changes that are making a difference in the care that we deliver. So all of us in this field of geriatrics and gerontology understand this concept. I don't think I need to explain this too much, but I was struck by one of these quotes, which says, ageism is one of the last socially accepted prejudices. And you know what I mean. I think I don't have to go into this, right? In fact, as geriatricians, we still have to explain to our colleagues sometimes why geriatricians are necessary. As UCI experts in elder abuse, we also experience firsthand the difference in how elder abuse is treated compared to child abuse and domestic violence. So unfortunately, ageism is still something that we have to be aware of. How does ageism affect us in healthcare? Well, in this case, ageism strips us of the ability to see the everyday resilience and possibilities for resilience in our patients. In order to be good doctors and good clinicians, we need to understand our own biases and prevent that from affecting how we should practice. Because when we fall for ageism, we perpetuate the beliefs against our future selves. So there's an article here from Kaiser Health News, which gives some examples about how ageism affects healthcare. The thought that maybe our patients can't recover from a treatment, so maybe we don't provide that treatment. That pain is a part of life, so maybe we don't treat as aggressively. That older people die. That loneliness is common. And that sometimes older people can't make their own decisions. There's also a huge financial cost to ageism, and there's some impressive research being done to show us this. There's a $63 billion cost of ageism by researchers who've done some of this, which amounts to one of every $7 spent on the 8 most expensive health conditions for all Americans over the age of 60 in 1 year alone. So among these health conditions imagined are the more common ones: cardiovascular disease, mental disorders, and chronic respiratory disease. Dementia is not a part of that group. So if you see this graph, you'll see that the types of ageism that were examined included the unfair treatment of older adults, as well as 2 categories of beliefs that are learned from society: negative stereotypes about older people and negative views of themselves as older individuals. Now, how does dementia fit into this picture? Well, we see that dementia is the core of ageism. Owing to mostly the negative social perceptions of dementia, people with dementia and their families often find themselves in very difficult situations. We need to eliminate the widespread social stigma to promote good health. And we know through the years, from Greek philosophers to the Middle Ages, that philosophers and theocrats have persisted in negative connotations that have persisted now in our modern times. And in many cases, these thoughts and beliefs prevent our patients from getting the care that they need, delay in diagnosis, and delay in treatment. So thanks to the Alzheimer's Association, we do have some data to back this up. Based on a 2015 report, only 45% of patients diagnosed with dementia and their families recognize dementia as an illness. Compared to cancer or cardiovascular disease, the understanding of dementia was significantly poor. Thanks again to these surveys, we see that only 24% of respondents, actually 24, hid their history of diagnosis of dementia due to social stigma, and they experienced denigration and rejection of their ideas when their illness became known. So there is a profound impact because of the social stigma and how people ask for help and how they expect to receive help. So the practice of medicine, now we're getting back to medicine, is influenced by society and our cultural norms. So how do we address this? And how do we transform our culture? And so I'm going to talk a little bit about the UCI way because we're addressing this head on. First of all, our belief is that we need to mandate screening for dementia as a standard part of healthcare prevention. This is currently not the case. Imagine and consider dementia is one of the main causes of disability in later life, ahead of cancer, ahead of cardiovascular disease, and stroke. In Orange County, it is one of the top 3 causes of death in older adults. We screen for heart disease. We have multiple screenings for cancer. Why not dementia? Now, we know that national consensus expert panels are very important for the delivery of care, but yet in the situation of dementia, we find that one of the main professional societies for primary care, United States Preventative Task Force, still holds and says there are insufficient evidence to screen for dementia. And remember, primary care is the front door to public health. Okay. Now, the Academy of Neurology does advocate screening for dementia over 65. And we do have governmental payers, Medicare and Medi-Cal, which advocate annual surveys for dementia. But not all of our organizations have caught up to this. But I think we should consider not the preponderance of evidence, since they can't seem to find the evidence to mandate screening, but let's consider perhaps the widespread harm of not screening. Do no harm. So we know that 1 in 2 people with dementia are victims of some type of elder abuse and neglect. We know that diagnosis is often misunderstood, and misunderstanding among families tears families apart, creates inappropriate communication and strains relationships. Let's not forget safety, the risk of self-neglect. Let's not forget the lost time that we could inform, educate, and support. And let's not forget the time that we could help our patients manage comorbidities like diabetes and hypertension and so forth. Okay. So for us, there's a lot of reasons here. Now, fortunately, we live in California, most of us here, I think. California tends to be a bit more progressive than a lot of states. We have been participating for the last 18 months in a program initiative from California called Dementia Care Aware from the Department of Healthcare Services. UCSF has led this charge, and we have partnered with them in addition to other UCs across the state. So our work in the last 18 months has been to train primary care healthcare providers in the screening of dementia. This is our group. So a very formidable geriatric group here. And I'm proud to say that we have trained, since I wrote this slide, Patty Mouton, and I want to mention that Patty Mouton and Alzheimer's OC as well as Alzheimer's LA have been strong partners with us. When the slide was written, we had trained over 1,800 healthcare providers in LA and Orange County in the last 18 months. I just ran into Patty in the hallway, who now tells me that the number is now 2008. So yes, we've have a formidable group and we've done a fantastic job. But it's not enough, right? In order to become standard of care, one thing that we would ask all of you is to advocate at the ground level that you receive this type of patient care. Okay. All right. This leads me to talk about our workforce. Why is it so important? So primary care, as I mentioned, is the front door to public health. It's really important now to understand the workforce landscape today and then in the next couple of decades. So if primary care is the front door to public health, just consider that by 2036, there will be a projected shortage of 68,000 primary care physicians. There will be shortages of geriatricians, neurologists, and geriatric psychiatrists. There will be a 50% increase in demand for geriatricians by 2030. In 2025, next year, the need for a neurologist will exceed our supply by 19%. And I will mention at this point, since I am a proud anteater, that we do have the only geriatric fellowship here in Orange County, and we graduate 3 geriatricians that stay and work in Orange County here to help our older adults. All right, so again, what can you do at the ground level? Anytime you can, advocate for education, advocate for training, advocate for more healthcare professionals that will address this field. Now, moving on to some of the positive changes that are happening. So there's a national movement afoot to talk about age-friendly health systems. There's an initiative by the Institute for Healthcare Improvement and the John A. Hartford Foundation. We're proud that UCI is a designated age-friendly health system. What does that mean? It means that we strive to address the 4 M's for every single patient, older patient in our hospital and our clinics. The 4 M's are here, you'll see them. I won't go into them in detail, but it's basically what geriatricians do, right? We want to address, first of all, what matters to our patients. Like, what is your goal? What's not my agenda? What is your agenda? And let's work together to achieve that. Next, we took a serious look at medications. We know that medication and polypharmacy are a very, very challenging problem. We have many patients that come to us with lists of medications, and one of the joys in our lives is to deprescribe instead of prescribe. So we take away as many as possible. We also look at mentation. So that means, again, we pay special attention to screening and the care for patients with dementia, as well as their caregivers, depression, anxiety, and other mental health disorders. And finally, we look at the mobility. Keeping active is so important for health. Right? So we want to understand and make sure people can continue to move about. And all of this works together so that patients can remain and maintain their independence in our communities. Okay. Now, what do we need for ideal dementia care? Along with these amazing scientific innovations at Mind and elsewhere, what do we need? We need models of innovative care with team-based care. I'm proud to say that at UCI, we've had the support over a number of years to develop these teams. For us, primary care, geriatricians, neurologists, psychologists, social workers, pharmacists, physical therapists, nurses, and healthcare coaches. And many of them are under one roof in the Senior Health Center, and many of them are under other roofs in UCI, and we're working always to try to put them together. Okay. Next, I think what you've all been waiting for is the guide program, so I'm going to get there. All right. All right, so finally, you know, with the age-friendly health system, with the understanding that team-based care is really important, that one doctor, one physician, one person in a clinic cannot do all of this, we have the guide program. The guide program is a new program. It was first designed or announced last year. It's administered by the CMS for Medicare beneficiaries. The purpose of the program is to improve quality of life, to enable people with dementia to remain in their homes and their communities. It's a comprehensive and coordinated care. And of course, one of the main goals is to reduce strain on caregivers. Okay. So UCI, because we had the infrastructure already, applied for this program. Some programs, a lot of organizations applied for these programs. If they didn't have the infrastructure in place, they were admitted as a new program, and UCI was admitted as an established program because we already had a really good infrastructure in place. So we were accepted, and I'm proud to say that we just launched the program in July of 2024 with our first patient, and I'll explain what this means. And I will note that at UCI, patients need to have a primary care physician at UCI, and that's because we need to make sure there's 24/7 access to us. And the only way we can do that is through our 24/7 call system. Who's eligible for guide? So a beneficiary or a patient can receive services if they have a confirmed diagnosis of dementia, that they have Medicare Parts A and B, unfortunately not Medicare Advantage or not managed care, is not on hospice, and is not a long-term resident of a nursing home. The patients can self-refer or can be referred from their physicians. Now, a patient with or without a caregivers can be referred to the program. Caregivers are relatives, but they must be unpaid caregivers that assist with daily activities, whether it's basic daily activities or instrumental activities of daily living. If a person has a mild or moderate stage of dementia, they do not need to have a caregiver. But if someone has moderate to severe stages of dementia, then they need caregivers to be part of the program. And one unique part of this program is that there are respite services available. And CMS says that we must offer in-home respite and may offer adult day centers and facility respite. So for UCI, we're partnering again with Alzheimer's OC for the in-home and the adult day center respite options. So what are the benefits? So first of all, care navigation. And again, at UCI, we retain our geriatric specialists. We have a physician assistant with over 20 years of dementia care experience. And Valerie George is right over there. Okay. And she is going to be the core of this program. So if you have questions, please find her. And she'll be working with Dr. Sonia Segel, who again has been a geriatrician here for about 20 years. So we have a lot of expertise. Patients would undergo a comprehensive assessment to begin with, which leads to a personalized care plan, including a home visit to just check out the environment. And one nice thing about the program is we just don't accept you and let you go. We check in, whether it's monthly or quarterly, with every patient and caregiver. And then if there is like an urgent care visit, an emergency room visit, or hospitalization, those check-ins will become much more frequent to make sure that people have what they need for transitions of care. Now, I mentioned the respite for caregivers of moderate to severe persons living with dementia in collaboration with ALSOC. So CMS for eligible participants will provide up to $2,500 a year for the respite services. In terms of support and communication, we will maintain very close communication with primary care providers and clinicians and specialists. And I want to actually call out Dr. Sajati here at Neurology, who's been a very strong proponent of this and can't wait to start referring to us. So thank you, Dr. Sajati. And we will be providing education and support groups and so forth to our beneficiaries or our patients. So again, we've already accepted one. We've done a lot of work. We've had to integrate our electronic medical system to do this, integrate many things into the system. I won't bore you with that, but it's taken months to really prepare for this moment. And I thank our team for doing that. And we're very, very excited to be able to offer guide to our persons living with dementia and caregivers. All right, so to close, I just want to get back to the heart of the matter. What matters most? What matters most to our caregivers? So with all of this, we get it. We get that caregivers need understanding, support, and respite. We get that caregivers want to know they're doing everything possible and leaving no stone unturned for their loved ones. And how do we see you? We see you as resilient. We see you as problem solvers, selfless and strong. Now, what matters most to persons living with dementia? Our commitment to you is that we understand what matters to you and we help you make that happen. Imagine that it's to be acknowledged, that there's safety, there's security, acceptance, friends, understanding, and that we understand your goals of care. So we are here for you. So I'll end with this a little bit too fast, right? Okay, I'll end with this. You know, you can't go this alone. We know that. I mean, I think everybody in this room knows that. But I like this quote. It says, "I bend, but I do not break. But together we will bend, but we will become stronger. And together we are resilient." So please work with us. We want to be part of the community, and please work with us to become resilient and care for our persons living with dementia and caregivers. Thank you.
Take one or two questions.
Whoa. I like that question.
That's a good question. Yeah, I knew the answer.
Yeah.
What about the states that don't have any, not even a fraction of this infrastructure? They're cutting back on, I know you said Medicare, but
Yeah.
Medicaid, healthcare deliveries infrastructure is disappearing. They have to even leave the state. We talked, Dr. Robert talked about the genetic testing is not even existing in other states. So you've got big medical care deserts, and they call outside the state to this. How are we going to shore up those deserts opening up?
Well,
It's okay. I'm going to repeat it for the people online.
You got it.
Which is, what about the people who live in states that don't have the same infrastructure?
Well, I think, first of all, we can be models of care here in California. We're very fortunate. I mean, I think we are very fortunate to have what we have. But we need to be models of care. I think all of us can contribute to these advocacy organizations because they're in Washington, they're everywhere, you know, trying to do their job, trying to raise the voices of dementia. So all of us can contribute, volunteer, contribute to any of these organizations that are here to support us today. In terms of geriatrics, we have had many virtual education sessions open to the entire country. We've had audiences from almost a majority of the states, including Canada and other parts of the world. So we are training essentially across the country. But, you know, I think it's advocacy. You know, maybe we can help people in other states become advocates. But I think a lot of this has to be grassroots. We're doing what we can. And at the School of Medicine, we can advocate for the training of healthcare clinicians that will be ready to care for our patients. But we need a grassroots, I think, infrastructure as well.
Thank you so much for a fantastic talk. Year in and year out, one of the most valuable parts of this conference is to hear from people who are living through the things that our speakers come and discuss in their slide presentations. This has over the years included people in a lot of different positions, ranging from caregivers to people living with cognitive problems to advocates and research participants. It's an incredibly important part of this conference. It is why we do the work we do, is to try to make a difference in the lives of patients and families. And we are always so very grateful to the very brave, generous, giving people who participate in these panels, and today is no exception. I am joined by three amazing women today for today's panel as part of this 35th annual conference. The theme of the conference is Advances in Care, and we'll hear about why these advances are so important, why more advances remain desperately needed, and how some folks are trying to contribute to making those advances. So we have a fair amount of time in our schedule for this. I am going to introduce by name each of our panelists. They're going to, in turn, tell you a little bit more about their stories and their journeys. Then we'll have a bit of a Q&A with me up here before we open it up for audience questions at the close. So again, I am personally just so very very grateful to all three of you for doing this, and so excited. I was telling these ladies they are faculty members in this conference, and I'm excited for our audience to get to learn from them. We're joined by Patty Jo Corrali, Denise McKeever, and Catherine McAdams. And so in turn, I'm going to let them tell you a little bit about their journeys to get us started. So Patty Jo, could you get us started?
Hi. Wow. My name is Patty Jo Corrali, and I am here for, I think, 4 reasons in my life. One is that I am a double ApoE4 carrier homozygote. The next one is that I was a caregiver for my mother with Alzheimer's as she passed away 2 years ago. And I brought my little visual aid here because this was the little stuffed animal that I would take in my mother's final 6 months or so that gave her comfort. And it made her feel that she had love in her life still, even though she couldn't process a lot anymore. The last 2 reasons are that I am involved in UCI Mind. When I learned of my double ApoE4 status, I did everything I could to get involved in some kind of clinical drug trial or anything, just to help not only myself if possible, but just to kind of get the ball rolling for other people after me. So I was accepted into the longitudinal study at UCI Mind about 8 years ago. I'm going into my 9th testing probably at the end of this year. And through that and working with Dr. Sajati, and when my mother passed away, I brought her brain autopsy results so Dr. Sajati could explain them to me, because it was too much medical terminology, and it took me a half an hour to Google even one of the terms. So Dr. Sajati was very kind and sat with me, and during that meeting with him, I got choked up because I said, I'm a double ApoE4. I'm already in your longitudinal study. What am I supposed to do? So it was a little bit of it makes me a little choked up now, because when you're in the situation, you've seen my grandmother passed away of Alzheimer's, my mother passed away of Alzheimer's, and I have genetic markers. I know genetics are not destiny. I'm doing everything I can to avoid that, but is there any help on the other side of this to help me as well? So he's took that. I don't think he liked to have somebody crying sitting in front of him. So he said, hold on just a minute. And he went and spoke with Robin, who worked, I think she recently retired from UCI Mind. And between the two of them, they were able to get me accepted into the clinical drug trial of Leqembi. And so I've been in that for 1 year now, and I feel really good about it. I don't know if I'm getting placebo or the real thing, but I don't care because it's better than nothing. And I really enjoy the people over at the AHEAD study and all the help that they give me and the comfort, and is sort of a friend. They're friends now in a way. So I brought this little visual aid because I wanted to say that as a family member of an Alzheimer's sort of legacy, I watched my grandmother die of this. My grandmother was a very shy person, didn't have a lot of friends and didn't want a lot of friends. She was an artist, a painter. She liked to stay home. She liked to take care of the house. She loved her husband and her children. And she was not stupid. She was one of the earliest people. She only a woman in her family who got a college education in 1928. She was on that track and was able to carry on wonderful conversations until she couldn't. And she went down the road of Alzheimer's, and it was incredibly sad. My mother, who was her daughter, was an accomplished musician, a near musical prodigy at a very young age, and went off to the University of Michigan and became a middle school chorus teacher for 35 years. She was also Woman of the Year in Laguna Beach. She started the community band in Laguna Beach. She was on the Arts Commission. She was on every single thing you can imagine. My mother had more friends than anybody should ever have. She was extremely social. She was very involved in everything. She ran her own finances. She also got Alzheimer's. My mother drank like a fish. My grandmother didn't drink at all. There's nothing that I can see between the two of them that would have caused both of them to look nearly like twins in their later years because of Alzheimer's. My mother was a round person. She always carried an extra 50 pounds. My grandmother was a slender person. There was nothing about them in their lifestyles that was similar except their genetics. And then that came to me as well. So you can imagine that when it comes time to me and now it's my turn, what are you going to do? You turn to actual medicine, the real thing, not the pseudoscience. People come to me all the time. You should take magnesium. You should take this. Have you seen this doctor? Have you seen this doctor? Did you watch this CNN special? Did you? People are constantly bombarding me with those things, and I turn only to the real science, and that's where I am now. And I'm hoping that through that avenue, there's help. There's help for me. And the early diagnosis is really important. I was telling the table at lunch that my mom was scammed on the phone when somebody called her and said, "Hey, Grandma, I'm in a Guatemalan prison. Can you wire me $10,000?" And she did. She wired not my daughter, but she thought it was, and she didn't do any checking. She sent that 10 grand off and it was gone. If some kind of diagnosis had happened at that point where I could have gone to the bank and said, "Don't let my mom do any of these things," that would have been very helpful. So it's all with you guys from now on. Thank you.
That's fantastic description. Thank you for everything you just said. The AHEAD study is a study that Dr. Pierce mentioned earlier in her talk. We saw the data for lecanemab and how impressively it can lower amyloid levels in the brain of people with mild cognitive impairment or mild dementia. The AHEAD study is testing whether lowering amyloid levels in people who don't have mild cognitive impairment or dementia, that is, they have normal memory performance. We're testing if doing that can stave off, delay, or prevent altogether the onset of memory problems. It's a major, major undertaking. There's a similar study with donanemab, I should say. Both are incredibly exciting. Both have completed recruitment. And so we're about 4 years away from getting the answer to that exact question. And that too is incredible progress. There's also, we didn't talk about it much today, there's also remarkable data about the financial errors and susceptibility to scams that people with cognitive impairment deal with. And numerous studies have shown that financial errors, mortgage defaults, other things are much more likely in people who will someday get a diagnosis of dementia. So we know that these challenges begin very early. We don't have enough as a society to protect older people, like Dr. Gibbs was saying. And of course, people who are developing cognitive problems are at the absolute greatest risk for falling prey to things like these terrible scams. She sounds like a great grandma, though. Okay. Denise, could you tell us a little bit about your journey?
Hi, is this working? Yeah. I'm Denise McKeever. I am the care partner for my husband, Joe. He's 72, and he was diagnosed last year with Lewy body dementia. My story, we heard earlier how there are communities of people that have trouble getting diagnoses. It's harder for them. And my husband doesn't fit those communities. He's a white man. He has means. He lives in Orange County. He practiced law for over 30 years. He had a long and successful career, very intelligent, highly educated. But my story is it's still hard to get a diagnosis. And I'm not blaming the medical community. It's a complicated issue. Joe had, well, let's see, he had a long history of anxiety and depression, which ran in his family. When he was 65 in 2016, he was having trouble at work. It was stressful, moving too fast. He'd call me and say, you know, they were talking about something in a meeting and I just wasn't catching it. I just, you know, he was overwhelmed. So he, without really a lot of planning, he retired pretty suddenly because the anxiety was too much. And by that time, and I don't know when the symptoms started, but by that time I know he was having restless leg. He wasn't sleeping. He was yelling at night. But I mean, we didn't know anything about Lewy body, and I don't know if those are the early signs of Lewy body, but people kept saying, well, if you only get some sleep, you'll be better. You know, there's all the answers. So we started looking for answers. We went to a psychiatrist and he said, well, you have a type of bipolar, type 2, so we'll treat you for bipolar. We went to a neurologist and, you know, he did the MRI and the muscle test and he did all the tests and he said, you know, I just don't see anything wrong. I think it's just a psychiatric problem. So, and we went to a sleep specialist because of the restless leg being so severe. He would be up walking the house all night. And there's really not a good cure or remedy for restless legs, so we were struggling with that. Our primary care physician sent us to a neuropsychologist. So we got the cognitive testing and the report came back, yeah, you do have some cognitive issues, but you had a traumatic brain injury 20 years ago when you had a bicycle accident. So maybe that's just manifesting now as you age. We think you should eat a Mediterranean diet and exercise. And that's good advice, but we were already eating an organic, whole food, plant-based diet, and he's an accomplished marathon runner. So not much else we could do in those counts. So, you know, there we were, just, and he was tired of testing, you know, sleep studies, MRIs, all that. He was tired of everything. He didn't want to go to any other doctors. He was having back trouble then, a lot of pain. So it was easy to say, oh, your problems are because of the pain or the pain medications. And ultimately he had surgery for his back. It was pretty intense. It was a triple fusion in his lumbar spine. And it, when he came back into his room after recovery, he was in a state of delirium for 3 days. So his cognitive abilities took a big hit. And coincidentally, right before his surgery, or a few months prior, I had convinced him that we should give one more doctor a try, that we weren't at the end of the line, we should still try one more time to get a diagnosis. So he called UCI neurology and he said, well, I don't know who to go to, but can you just get me an appointment with a neurologist? And, you know, as luck would have it, we saw Dr. Sajati. And he was able to almost immediately suggest that it was Lewy body. Now, whether that's because his symptoms had progressed enough in those several years that it was more evident, or, you know, I don't know. When I look back at the self-reported symptoms when he did the neuropsych test, he was having hallucinations. He was already, this test showed he had a impaired sense of smell. He already had a lot of Lewy body symptoms, but no one was seeing it. And maybe because he had, you know, anxiety, depression, he had traumatic brain injury, you know, so it wasn't clear. But anyway, that brings us to today, and we're living with Lewy body. Mm-hmm.
So I would say, unfortunately, this too kind of fits with some of the things we heard today about the challenges that people can encounter finding someone who understands these disorders in the older person and the older brain. You told me a little bit about, you know, your husband was obviously extremely high functioning. I think I remember you telling me that even when he retired as a lawyer, he started doing pro bono immigration law. And learn Spanish to boot. And a fundamental part of Lewy body disease is cognitive fluctuations. And so, if people really don't know what to look for, it's easily missed. I guess another common theme so far is Ahmad Sajadi's expertise on our panel, which is great to hear. But, I think that we heard, in several talks today from Dr. Schindler and Dr. Gibbs about, the dearth of folks with the skills to make these diagnoses. And while the tools are coming so fast to help and support those diagnoses, I think it's really important to say right now, we're nowhere near not needing those expert clinicians to arrive at this. And the tests on their own are not going to be enough anytime soon. They may be extremely valuable for screening programs, but right now the role of the expert clinician really cannot be overstated. Our last panelist is an expert clinician. She's a provider of care and a care provider. Catherine, could you tell your story?
I have a little different story. My name is Catherine. I have been one of the nurse practitioners at UCI Mind. I've been there for about 30 years. And I have had the privilege to walk with patients who are cognitively normal, who have questionable cognitive impairment, who have mild cognitive impairment, who have Alzheimer's, Lewy body, FTD. Anyway, I have walked with hundreds and hundreds of patients through their journey and have gone to many, many funerals, because by the time they go on to the next world, I know their families very well. It's been an honor to be here for so long. And then my husband of 41 years was diagnosed by Dr. Sajadi as a frontotemporal dementia with a behavior variant. As he progressed through his disease, he developed excoriation syndrome, and now he also has some ALS symptoms. And most of my coworkers know a little bit about my situation, but for a lot of them, they have no clue that this has been going on. He was diagnosed maybe 3-4 years ago, maybe, but he has been showing symptoms since, I would say, 2016 or 17. And his first symptom was apathy and withdrawal. He would not go to any of our daughter's functions at schools. And then he just started doing weird things, taking off in his truck. I can't even count how many times, and drive around the block and come back home and watch TV. And then he would take 10 to 15 showers every day. And at first I thought, what is going on? And I thought, well, and I knew there was a dementia problem of some sort, a memory problem, but he was in denial. He said he had no problem whatsoever and to leave him alone. And it got really hard. And my family and his family did not believe me. His brothers still do not believe me today. And it's hard to take emotionally, but you just go on. You do a lot of prayer, and I've got many blessings through this whole process from my coworkers, from Caregiver Resources of Orange County, from Alzheimer's OC, and from Dr. Sajadi and Dr. Tai, who have been at my side the entire way. One day when I was at work, I was called by the Orange County Sheriff. And our neighbors across the street had been having some construction done in their home. And this one worker kept parking in front of the fire hydrant in front of our home. And my husband I guess, had had enough of this, and so he put the worker in a chokehold. Behavior variant, okay? And they called the police, and I was instructed to come home immediately from work. So I dropped everything, went home, and he was put under arrest. And I explained that he had dementia. They let him off, and he was released to my custody. From there, I took away his driver's everything. I took away the keys. I hid the keys in the house. He found them. He took off in his truck. And things just progressed from there. I would take him to farmers markets. He would say he had to go to the bathroom. He would come out of the porta potty with his pants down around his ankles. It got to the point where I could no longer take him to church because he was engaging with children. You know how children are in church, and they turn around and they look and smile. Well, he was doing the same thing back with them. And it's fine when children do that, but when an older man does that, there's a problem in the eyes of others. I put him into daycare at the Adult Day Healthcare Center in Laguna Woods, and he thrived there. And he progressed, and then he became doubly incontinent, and he was still at home with me. And one day he had an accident in one of the lounge chairs, and it took 4 people all afternoon to clean up after him. And on that day, I was asked to not bring him back. In November of 2023, I was scheduled for surgery, and the surgeon said, "You're not taking care of him because you won't be able to use your hands." So I placed him in a board and care near my home. And again, he's thrived there. His irritability has turned 180 degrees. He's the happiest, most oblivious person you'd ever want to meet. And they now have to use a lift to move him. He doesn't walk. He's gaining weight, but he's happy. He's on medications to control his behavior, and it's so far so good. And now he's on hospice, and I have made arrangements, funeral arrangements already, so that all is required is a phone call. And please, if you're a caregiver, please do that. And so I am so blessed and so happy to still be working here with my family and all my friends. That's it. Thank you.
So we're going to open it up, and maybe I'll just ask a few questions to keep us going here. So thank you all. Those are incredible descriptions of your journeys. I guess you've all been caregivers. And caregiving is a two-sided coin. It's obviously difficult. It creates burden, but many people find power and value in providing care to someone in a unique way. So I guess in turn, maybe if you could just talk about, both the most challenging part of your role as a caregiver, and we heard some of that, but also, how do you keep going? How do you find value in your role?
I was a caregiver for my mother who had Alzheimer's and passed away 2 years ago. And when I think back on that time, like I said before, my mother had a whole bunch of friends. And the call that, or the in passing comment that I always got that made me kind of mad was that, let me know if you need anything. That is like a get out of jail free card for a friend, right? Like I'm a nice person, but I'm not actually going to be there for you. And then the other one, I would say, if you want to be a help to somebody who's caregiving, just actually show up at the door. Just knock on the door and say, I'm here right now. I've got 15 minutes. What can I do? Can I help you clean up his accident, or can I help prepare some food? Those are the actual things that you need. And then because I'm very open and talk about Alzheimer's a lot and my personal journey through it, I hear people talk about their families a lot. Oh, my mother is having it now, or yeah, everybody has an Alzheimer's story. Every single person, even the Uber driver I took back home last night after the dinner, was telling me about her friend with Alzheimer's. And one of the things that really upsets me the most in that is when people say, oh, I don't visit my mother anymore because she doesn't know me. She no longer recognizes me, so I don't go anymore. And I feel like I'm not a violent person, but I feel like I want to kill them, because it's not now anymore that they give you love. It is your time to give them love. And all I can say is, you know who she is, right? And when you're a caregiver, my mom, I had to put my mom in a memory care facility, and showing up a lot for your loved one with dementia, especially in the final stages, they need to know that that person is loved by someone. That person is the loved one is going to show up and find out that their underwear isn't in their drawer, or they're not wearing their own clothes today. Where did these clothes come from? I mean, there's all kinds of those things where the person who knows the patient is the primary person now. The patient is now in final stages. Of course, they don't know you anymore, but now you show your love. It's now on you. That's kind of my soapbox.
Well, we all know caregiving is really hard, and oh gosh, I don't think very many people sign up for it. I mean, I raised a daughter by myself. I helped my father through cancer, end of life. I helped my mother through end of life. I finally, met Joe in my 50s and thought, we're going to have a good last chapter together. Okay, and I see. Okay, and I didn't know that, of course, I would end up being a caregiver yet again. I think Patty Jo is right. I don't have the bandwidth to tell someone what I need. What it really helps me is if someone says, if they're going to invite me to something, how about coffee on Tuesday at 10 o'clock? I can say yes or no. I don't have to figure things out. And I'd say one of the biggest challenges, especially early on, and if your person is pretty good at hiding, and Lewy body is really variable, so he could kind of hide some of the symptoms from friends and family. I could tell them with words what was going on, but they didn't see it. And words, even when I would tell close family members some of the more graphic things, they found it hard to believe. But they heard the words, but it wasn't until they came into the house and experienced it that they understood what the words meant. So I think now most of the family and friends are pretty much on board. They understand what they can understand. But I would say the gifts of caregiving, in my case, Joe he still functions really well. It's up and down, but he functions really well. He's very self-aware. He knows what's going on. And we've had some really emotionally intimate, important conversations. And, I've talked to some of my friends and they've said, gosh, I will never have that with my husband. We just don't have that connection. So for that, I'm grateful. And I just read something the other day, a concept that I'm thinking about, which I think is important for caregivers as well as, medical health providers. Consider that healing is possible even when a cure is not. So that's what I'm working on now. Thank you.
it's all challenging for me, I think. It's hard. And I truly thought I understood what caregivers had been going through when, you know, through work. I related to all the caregivers, but I didn't really realize it until I was walking myself down the same path. And now, when I give advice to caregivers or make referrals, it's coming from a combination of the heart, from science, from intelligence, and from knowledge that I've gained. But I think most of it is now coming from my heart and a true understanding of what caregivers are going through. And for myself, I don't usually ask for help, but I have asked for help from God, and He always comes through, always. And He has helped me carry on, and I want to live. My husband does not recognize me at least 50% of the time. It's hard, but it's a blessing because he's happy. He doesn't know what's happening to him. He doesn't know that he's on the highway to heaven. So it's challenging, but I'm constantly blessed. And I know that those blessings will continue.
So we're almost out of time, but we'll open it up for one or two questions from the audience.
Thank you. Can we give a hand to these ladies? What a wonderful. What you have gone through and what you're sharing with all of us. My journey, I'm a physician, so I may be one of those that missed the mark many times. But my mother had dementia as well, and she was in a nursing home for a few years. She went from Orange County to Arcadia because my two sisters said, we want to see her every day for lunch and dinner. So I could go every Saturday to feed her. And I did that for 5 years. And yesterday I wrote a poem. I write poetry. So I wrote a poem, and this might, just 3 lines of it, is that in her last days, she actually shared a light. She was not able to talk, but there was a light, a radiance in that room, heart to heart. And that I would always explain to my siblings what I felt when I spent those few hours with her on those Saturdays. So yesterday I wrote, Amma, which is mom, Amma's light radiant was important. Her silent teaching from her heart to mine, showing me the path with no words. So I think you are still being shown that without words, there is a lot that is still happening. So please treasure that as you shared all the treasures with all of us. Thank you.
Thank you. One question, you all have the benefit of 2020, sites. Knowing what you know now, would you have done anything differently early on? And can you share that for those of us who are in it too?
I would not have given up when I was denied help from the VA. My husband's a vet. I still work. We don't qualify. I guess If you need help somewhere and they close the door, keep knocking. Keep knocking. That's what I can say. Don't give up.
Everything. There's also one question from the online group. Okay. My wife of 40 years has been diagnosed with FTD after numerous neuropsychiatric tests, PET scans, lumbar punctures, and MRIs. Her current neurologist at UCLA is at UCLA, and I would like to have my wife participate in UCI's guide program. Please let me know how we can do that.
I think we'll have to take that one up afterward. We'll connect that person. Val, do you want to say anything about further?
UCLA has also been approved for guide. So they just need to look into either from their primary care or neurologist and be a referral for guide program.
So I think what we're hearing is everybody needs more help. And I agree. Yeah, please.
I was going to answer the question because it made me think about what you would do differently. And I think when you're in it, you're just every day fielding balls, right? They're just coming at you and you're doing the best you can. And if you start to doubt yourself, you'll miss a ball or two or three. And so you just got to keep swinging. And I think there's not a lot of retrospective grace for that because it's so hard. I guess the only thing I could say is on a social level, it would be nicer if there was a better awareness of what Alzheimer's or any of the other dementias need in their daily lives. From all of us, from the whole community, from us as a society, from us as a friend network, from us as family. There's would be a better awareness, I think, if we all could look at the Alzheimer's or dementia patient with a sense of friendliness rather than a sense of, oh God, get me away from this person. I don't want to be drawn into this. Because it's really more of a community-wide effort that's really needed at this time. We all need to take care of each other, and a dementia person needs as many people as possible. So if you have somebody who's going through it and you know their family member, do something.
Also.
And I want to acknowledge that I feel really fortunate that when we had our diagnosis, people fell into my life that I didn't even know I needed. I was really lucky and, because of where we live and the resources we have here, I was directed to, Alzheimer's OC, and I just found, support groups and new friends, and all of that is critically important. And it's just a lead on what you've just said, Patty Jo, that, yeah, it's people. And I found friends don't know what to do because they don't know what it is. So there's a lot of education that has to happen for the community at large.
Yeah, we're very lucky to live in a resource-rich area. And we heard today about, not every part of the country is as fortunate as we are, or even every part of the state. Catherine?
Are there caregivers in the room? Okay, I've always said, do not neglect your health. There's a 50% chance that the caregiver will die first. I neglected my health and somebody woke me up. So please, please take care of your own health. It's kind of like that oxygen mask in the airplane. Put it on yourself first and then put it on the one you're taking care of, because that's how you're going to survive.
So we've got 3 Women of the Year up here. Let's give them one more round of applause. Thank you all for telling us your story, sharing your learnings, being faculty members of this year's conference. We hope you enjoyed this 35th annual conference. It is a time of a lot of exciting progress, but a lot of work left to be done. So join us next year for the 36th. We'll catch you up on what we get done between now and then. Thanks very much.